Hello everyone, and welcome to the 100th episode of the Biologic Podcast. I'm going to do something a bit different and tell you right here at the start of the show to hit that like button and subscribe to the channel. It really helps me out, and you get more awesome biological content in your subscription feed. Now, it's the 100th episode, and to mark the occasion, I wanted to do something a bit different. Something a bit out of the ordinary, while still keeping to the theme of the series of these early metazoans. Because I usually cover living things, or lineages that are alive today, I often skim over many of the interesting things that have gone extinct, tens or hundreds of millions of years ago. If you can see where I'm going with this, for this special 100th episode, I want to explore the earliest prototypes of animal and animal-like life. I want to explore these particular macroscopic multicellular heterotrophs and their ecological communities that were among the most radical and complex organisms ever produced by the forges of evolution to that time. These primordial organisms existed in the Ediacaran period, a time span lasting from 635 to 540 million years ago. In other words, I want to explore the roughly 100 million year long period of time which saw the emergence of the Ediacaran Biota, with an emphasis on the larger animals that were evolving at the time. If you've been listening to this series on the early metazoans, then you should be familiar with the evolution of multicellular animal life, of poriferan sponges and ctenophore comb jellies. I've dropped tantalizing hints throughout about jellyfish, but that will be the topic of the next episode, not this one. Rather, today we're going to take a step back and look at the entire time period in which these first strange animal lineages came about, as well as all of the other weird animal-like creatures that also existed at the time, in this Ediacaran period. This was a time immediately before the Cambrian period, before the Cambrian explosion itself, some 538.8 million years ago. The Cambrian explosion was an incredible evolutionary diversification event that spanned a couple dozen million years. This event was a time of great change, because many new lineages of animals were being established, including the ancestral lines of all modern animal phyla. As a quick aside, the phylum is a higher-order taxonomic rank, immediately below the kingdom, so think like fungal kingdom, plant kingdom, animal kingdom. Within the animal kingdom, there's about 40 major subdivisions, or phyla. One of the more significant phyla is Chordata, or the chordates, which includes all vertebrate animals like you and me. The Arthropoda and Mollusca are both phyla that emerged in the late Ediacaran or the early Cambrian, and the Porifera, the Ctenophora, and the Cnidaria are each their own phyla that had already been around for a while by the time these other ones emerged. The rest of the animal phyla are generally small, obscure groups, usually extinct, and among those that still exist, at least a dozen of them are various types of worms or worm-like creatures. If you want to learn more about them, then go listen to episode 85, A World of Worms. In addition to this new stock produced in the explosion, there was an entire marine ecology of aquatic animals that already existed before the Cambrian explosion. In fact, the Cambrian explosion wouldn't have been possible without the proliferation of multicellular animal and animal-like life in the Ediacaran. It's just that when the Cambrian period came about, the new animal forms outcompeted the old ones, or altered the environment in ways the old forms couldn't tolerate, and many of them died out, either relatively quickly and calamitously, or in a dragged-out process, where their populations slowly dwindled to nothing. There are some survivors from this time, from this earliest evolutionarily workshopped generation of animals. There's the sponges and the ctenophores that we've already taken a closer look at, but there's also the cnidarians, like the corals, the anemones, the hydra, and the many different kinds of jellyfish. However, these creatures, whose lineages are still alive today, represent but a small fraction of the total life that existed in the Ediacaran. All the rest was wiped out by the Cambrian generation of animals. It was an ocean-spanning, or perhaps more accurately, a planet-spanning zoological turnover event that made the world much more familiar to what we know today, with arthropods and mollusks and vertebrate fish. The Ediacaran world, in contrast, was full of strange animals with weird body plans and a slew of unrecognizable features. They lived in a different world, in a world without bones or claws, without jawed predators hunting around, where life was thriving after the end of the most extreme ice age in the planet's history, and where almost any animal and its neighbor in the rock crevice next door were likely soft-bodied creatures with a body shape and a life strategy that would seem almost alien to us. In this way, the Ediacaran represents a lost world, or an alien world of sorts. Or to put a more positive spin on it, the Ediacaran could be seen as a young world, or a nascent world, like a metaphorical embryological stage of the entire animal lineage, which would come to diversify and dominate the planet's ecologies for hundreds of millions of years into the modern day. In today's show, we're going to explore this long-gone alien world that once existed here on Earth. We're going to explore the Ediacaran Biota and try to reimagine what these primordial marine communities and the organisms within them would have looked like. To begin this journey into the ancient past, we find ourselves in south-central China, in a relatively rugged landscape around the Yangtze River that at one time, hundreds of millions of years ago, was entirely submerged. But the water wasn't very deep, as this area was continental shelf, with a shallow water column that let sunlight touch the sandy sea floor. Photosynthetic cyanobacteria and algae were abundant. This particular habitat also happened to be rich in fine mineral grains, like phosphates and silicates. There were several reasons for this. First, before the Ediacaran, there was a period of extreme glaciation that gripped the planet for about 90 million years. In great natural cycles, sheets of ice moved towards the equator in pulses, and then receded, and then back again. This movement of glaciers all over the planet created extensive fields of gravel and finely ground sediment. However, the extensive ice coverage and the preponderance of snow as opposed to rain meant that these sediments were not readily displaced by water. They weren't washed downstream. These sediments and all of their mineral nutrients were largely locked on dry land, bound up under the ice. But when these ice sheets receded and this age of glaciation came to an end, a reawakened water cycle began to wash these sediments into the water. The second reason is that at the beginning of the Ediacaran, there really weren't many creatures that made any type of mineralized skeleton, so there was relatively low ecological demand for inorganic resources like this. Yet another reason is the increasing abundance of oxygen from booming populations of cyanobacteria and algae. This oxygen enriched the oceans, which would have made organic reactions with phosphates more likely. All of this oxygen, in combination with the phosphates and silicates abundant in both water column and on the seafloor, it created the ideal conditions for extremely high detail preservation of dead organisms. This was particularly true for cells with stiffened cell walls, like bacteria and fungi. Their wall structures were quickly replaced with calcium phosphates, and were thus preserved like geological flash photography. This amazing sediment-rich habitat that makes these super high-quality fossils is now known to us as the Doshanto Formation. This was once an ancient shallow-water continental shelf ecosystem, enclosed to the south by a carbonate shoal complex that turned the entire shallow-water area into a coastal saltwater lagoon. In the millions of years since, geologic forces have raised up the area, drained out the water, and wrinkled it up into a mountainous landscape in south-central China. Exploring this rugged landscape reveals to us a section of seafloor from 635 to 551 million years ago, including the fossilized remains of both deep-water, low-oxygen habitats and more oxygenated shallow-water shelf habitats. To put it another way, the Doshanto Formation, and the more specific regions and layers and structures within it, is like a geological documentation of the first 90% of the Ediacaran period. It tells us an incredible story of the evolution of multicellular life. There are 3 primary layers in the Doshanto Formation, corresponding to periods in the early Ediacaran, when the glaciers were still melting and the water levels were still rising substantially over time. The uppermost, or the youngest layer, is the most interesting for our search for life, but the older layers contain important fossils and isotope data that have given us some valuable insight on this understudied time period. There's evidence of an Earth that was actually rather hostile to life. The Ediacaran period itself began with the end of a massive ice age that saw the Earth almost or totally covered in snow and ice. At the end of this ice age, there was a radically changing climate to go along with the collapse of the ice sheets and the retreat of the glaciers. These environmental conditions were slowly improving over time, like with more oxygen and more nutrient influx from the washed-out sediment on dry land, but it was still rather unfriendly. For instance, there appear to be alternating periods of relative anoxia and oxygenation, as well as periods of widespread exposure to hydrogen sulfide, perhaps from heavy volcanic activity. Fossil traces of multicellular life are virtually non-existent in the older layers of the Doshanto Formation, but there's extensive fossil evidence of microbial mats and chemical evidence of microbial metabolic activity. One interesting exception to this are fossils of filamentous microbes from the base of the formation, dating to 635 million years ago. These curious filamentous structures are thought to be hyphae from a fungus, and their presence here in this shallow water habitat has been interpreted as evidence of a fungal lineage in the process of adapting to and colonizing dry land. So before we get to any of the animals and the animal-like creatures, we have fungus doing its thing in the background, slowly moving closer to the shore. In time, these fungi would adapt to dry air and dry land conditions. They'd spread out over the mineral bedrock, growing into cracks and crevices, and releasing chelating chemicals in their saprophytic search for sustenance. But this is a process that would take hundreds of millions of years, well into the Cambrian and beyond, operating quietly in the background, as larger, noisier things, like plants and animals, would evolve later to dominate the terrestrial ecology. Coming back to the Doshanto Formation 635 million years ago, we come to something that, well, there's still some debate about it. But it appears that there was a biologically driven surge in oxygen production in this post-Ice Age, early Ediacaran time period. This is when we're seeing more and more nutrient runoff from the thawed-out, ice-free land surface, and algae and cyanobacteria populations are booming as a result of all of the dissolved iron, phosphorus, calcium, and whatever else. These microbes are photosynthetic, and as they photosynthesize, they produce both sugars and molecular oxygen. There's chemical traces of a vast pool of dissolved organic carbon in the early Ediacaran oceans, which was somehow oxygenated at a planetary scale, and the total oxygen levels kept increasing at an unprecedented rate. Now you might be wondering, didn't photosynthesis evolve 3.5 billion years ago? If photosynthesis has been around that long, why are we only seeing oxygen levels start to rise really quickly around 5, 6, 700 million years ago? That's a great question, and the answer is pretty remarkable. Molecular oxygen is very reactive. It likes to bind to all kinds of things. It can be dissolved in certain liquids, like ocean water, and it can be bound up in solids, like rocks. Most minerals are composed largely of silica and oxygen, with some other fun stuff mixed in. This means the mineral structure can incorporate oxygen. The mineral exposed to oxygen gas can literally absorb the gas as it gets integrated into the molecular structure of the mineral. Newly formed rock, like that spit out of a volcano, or subterranean rock lifted up to the surface through geological processes, it spent the last who knows how many millions of years in an anoxic subsurface, subterranean environment. But when it gets pushed up to the surface and exposed to the atmosphere, it can absorb huge quantities of oxygen. Of course, the oceans and the rocks can only absorb so much oxygen. Crystal matrices get completed, and there's no more room for new oxygen atoms to be absorbed. And ocean water gets saturated with oxygen to the point that gas diffuses out of the oceans at the same rate that it's absorbed, also known as equilibrium. These ancient algae and cyanobacteria photosynthesized for about 15 million centuries, or about 1.5 billion years. And almost all of the oxygen they produced in that time, in those 15 million centuries, was sucked up by all of the world's rocks and ocean water. While atmospheric oxygen was still pretty low, some of it was converted into ozone via exposure to lightning and UV radiation from the sun. This created yet another oxygen sink, one higher up in the atmosphere, the ozone layer, that formed over millions of years. But as with the rocks and the oceans, it too reached a point of equilibrium, of homeostasis, where the ozone layer itself slowed the production of new ozone, and the rate of ozone depletion eventually came to match its rate of creation. It wasn't until all three of these massive oxygen sinks were satiated that free molecular oxygen began to rapidly accumulate in the atmosphere. This acceleration began roughly 700 million years ago, and it continued throughout the Ediacaran, leading to a permanently higher level of oxygen in the atmosphere. It reached its highest levels in the Carboniferous, around 33 to 35%. But today, molecular oxygen is about 20% of the atmosphere. For comparison, during that 1.5 billion year period where the oxygen sinks were being slowly saturated, atmospheric oxygen levels persisted at around 5%. So this relative surge in available oxygen is believed to have been a major driver towards the increase in complexity of multicellular forms. We begin to see the evidence of this in the youngest, uppermost layer of the Doshanto Formation. Keep in mind, though, that all of these Doshanto fossils, even the multicellular ones, are microscopic. But there's an incredible degree of biodiversity here. We've already mentioned cyanobacteria, as well as the more complex eukaryotic algae, which comes in brown, red, and green forms, but there's so much more packed into this Doshanto Formation. For example, all kinds of ciliates have been discovered, which are curious single-celled microbes covered in cilia, which they use to swim around and bring food particles towards their mouths, or whatever orifice or absorptive surface they use to take in nutrients. Some of them have the classic oval or globule shape, while others are quite elongated, with worm-like or finger-like body shapes. Some even have a flared end opposite a tapered end, making them look like small trumpets or horns, or miniature single-celled snake-like things. There are multicellular fossils too, like thallophytes. These are extremely simple, rudimentary plants, which includes the algae I already mentioned, as well as the lichens, which are curious composite organisms, created by a fusion of plant and fungal symbiotes that integrate their cells and grow in a way unique to each individual symbiont. While many lichen bits are microscopic, this doesn't necessarily mean that the actual living organism, as it was in its natural habitat 500, 600, 700 million years ago, was microscopic. If a lichen is growing on a good, solid substrate, it can grow quite extensively, so it's entirely possible that the largest of these early Ediacaran lichens may have grown as buds or small patches on a rock that could have become large enough to be visible to the naked eye. There were also primitive seaweeds, which at the time were little more than sheets of photosynthetic phytocytes. These would have been abundant in a shallow water coastal habitat with ample sunlight, like you'd see in the Doshanto Formation. Although how large they would have grown, and in what sort of shape, I'm not sure. It's possible that they merely repeated a fractal pattern of branching, or they grew as a single linear strip, perhaps able to be seen with the naked eye as well. There's another type of microscopic fossil here in the Doshanto Formation that's particularly cryptic. These are the acritarchs. In a very general sense, acritarchs are small, spherical, or globular-shaped fossils, with a sort of shell of dense or harder tissue and a softer inner core that often fails to fossilize, leaving behind a hollow or a void space. This very general structure is about the only consistency in the acritarchs, and that's because this isn't actually a clade of organisms. Acritarch is like a catch-all term for any microfossil that matches this very general structural description. There are several groups of acritarchs that seem to be actual lineages of animal-like organisms, showing variation in traits consistent with evolutionary divergence over space and time. They appear to be eukaryotes, although among them there are both simple forms more similar to prokaryotes and larger, more complex, more traditionally eukaryote-like forms, with a wide diversity of appendages and features, like cilia or flagella, spines, flaps, scales, internal organelles, and so on and so forth. In fact, the simple, sort of prokaryote-like acritarch fossils can be found in sedimentary rocks that are over 1.8 billion years old, which means that they've existed way before the Ediacaran period. On the other hand, the more complex, eukaryote-like acritarchs appear much later in the fossil record, in the early days of the Ediacaran period, and they become particularly common in the uppermost layer of the Doshanto Formation. The fossils reveal an Ediacaran ecosystem that was surprisingly complex and dynamic. It appears that their populations crashed during periods of glaciation, and resurged when the climate was more accommodating, perhaps revealing an ecological association with photosynthetic microbes like cyanobacteria or algae. The acritarchs also show evidence of adaptations to defend against predation. Among these acritarchs were the large, ornamented Ediacaran microfossils, or to refer to them by an acronym, the loams. These loams existed in the early portions of the Ediacaran, and they disappeared from the fossil record by about 560 million years ago, when the rest of the greater Ediacaran biota really got going. Now, it might seem somewhat oxymoronical to call these things large microfossils, but that's actually pretty accurate. They are microfossils, they're microscopic, but compared to the other microfossils that existed before, during, and after them, they're actually quite big. They're around 100 micrometers in diameter. This is bigger than the dinoflagellates that came after them, and it's nearly 10 times bigger than the spores of the green algae that we see in fossils all around them. These loams are curious for another reason. They have a multi-layered outer wall, and there's discernible bits of internal anatomy within them, and ornamentation or growth processes on the outside. The purpose of these outer structures is debated. They take the form of spines or thorns, and the most obvious idea is that they're a defense measure, they're a defensive structure against predation. But we're talking about a time that's so long ago, that's so early in the evolutionary history of animals, that there may not have actually been predation beyond the activities of microbes and fungi. There may not have actually been any macroscopic or large microscopic predatory animals to evolve defenses against. Research on these loams and the purpose of their external ornamentation is still ongoing. Other defensive structures, like scales or plates or hardened body coverings, may have blocked the stinging cells or proboscis or probing tendrils of a predator. Now, as it stands, the acritarchs are a complicated mystery. They appear to be an ancient lineage, a really ancient lineage, which means that they're probably not animals. They may have been animal-like, but they belonged to an entirely different lineage that existed long before the most ancestral animals ever did. It's tempting to think of them as a massive group of aboriginal multicellular critters that belong to their own branch on the tree of life, with its own biodiversity and their own golden age of ecological dominance. But the reality is less glorious and more complicated. It's almost certain that there was a group like this, probably related to the algae, but it represents a fraction of all the known acritarch fossils. For example, there's a group of microbes called dinoflagellates, which are characterized by multiple flagella and a hardened outer surface composed of multiple layers of flattened membranes. These are reinforced in some species with a kind of cellulose-based armor plating. The dinoflagellates are classified as algae, as they descended from a primordial algae ancestor, making them a group of single-celled microbes closely related to the plants. Don't confuse them for the choanoflagellates, which are a group of single-celled microbes closely related to the animals. Anyway, as these ancestral algae were diverging and evolving into what would become the dinoflagellates, this involved the production of these hardened shell plates, or like an armor suit for a single-celled organism. When the dinoflagellate died and its body fossilized, the remains would resemble a sphere or a globule of hardened tissue with a soft inner core, which looks exactly like an acritarch. To make things more complicated, it seems that many, if not most, acritarch fossils probably aren't even organisms at all. They weren't even anything that was alive in the first place. On the topic of algae, some acritarch fossils appear to be clusters of algae cells, or the remains of the impressions their clusters left on the substrate. Furthermore, lots of them appear to be egg cases, perhaps from the earliest animals. The term metazoans refers to all animals, including those most derived and recently diverged, like primates or horses or something like that. But it's a term that's usually only used in a context when people are talking about the really primitive animals, like the sponges and the comb jellies and the jellyfish, and the even more primitive lineages that came before them but have since gone extinct. The tabulate corals, for example, existed after the Cambrian explosion, but they would have been the result, an offshoot or a divergence, of a lineage that went back millions of years, going back into the Ediacaran. And these ancient protocorals, these earliest of the metazoans, would have laid eggs. They would have released these eggs into the water column as an attempt at external fertilization. And inevitably, not all of them are going to get fertilized and turn into new individuals. Some of those eggs are going to get buried in the sand. They're going to get fossilized. We're going to find them and we're going to say, hey, look, we have a stiffer blob of tissue with a softer inner core. It looks like an acritarch. Also present around this time, 580 million years ago, would have been the earliest demosponges and the earliest cnidarians, like primordial anemones, which could also reproduce using tiny larvae hatched out of tiny eggs or tiny polyps. These small structures, be they larva, polyp, or fertilized or unfertilized egg, would fossilize in the form of spheres of harder tissue surrounding a softer inner core, just like an actual organism acritarch. Some of the most exciting acritarch fossils are interpreted to be animal embryos in various stages of development. Electron microscope scanning has revealed the shape of many of these embryos, clearly showing the bulges of growing and dividing cells in the surface of the young embryo. The uppermost parts of the Doshanto Formation also contain the fossil remains of adult sponges and cnidarians, which is conclusive proof that they existed by this time, at least 580 million years ago, and likely came from ancestral lineages that are thousands or millions of years older. I should point out, though, that even though these are larger multicellular creatures, including more derived animals like sponges and Cnidarians, even these fossils in the Doshanto Formation are microscopic, or at least really, really small. As a quick aside, the Doshanto Formation is enormous. It's this sprawling geological structure that exists all across central and southern China. Its fossil beds cover a vast period of time, including huge tracts of the early Ediacaran. But among this sprawling geological formation, there's also a few isolated beds that preserve some more recent creatures. And in the youngest, uppermost layers of the Doshanto Formation, or not even that, in the uppermost, youngest layers of very select sections of the Doshanto Formation. For example, there's a subsection or a sub-region called the Miaohe member, and the Miaohe member contains fossils, a whole mini biota, really, that are actually macroscopic, although these represent a very, very small fraction of the youngest slices of the entire Doshanto Formation. For the most part, especially when we're talking about these older, earlier Ediacaran periods, so before a lot of the Ediacaran biota proper showed up, before 560 million years ago, a lot of these fossils, pretty much all of them in the Doshanto Formation before this, are microscopic. Perhaps the most interesting fossil beds can be found in southern China, in a Lagerstätten deposit dated to be between 609 and 570 million years old, although many of the species fossilized inside of it are believed to have been in existence already for millions of years, and perhaps might be as much as 632 million years old. Now, this section of the rock in the Doshanto Formation contains a variety of mid-Ediacaran fossils known as the Wang-En Biota. Naturally, there's a lot of algae and larger phytocyte fragments, and lots of acritarchs, including a significant number of the embryo-like acritarchs. But there's also some weirder stuff that we can't really explain. For example, the Wang-En Biota includes tubular microfossils that are thought to be Cnidarians or Cnidarian relatives, although recent analysis has found their cells to be more like corals. But it's been suggested that they may be something even more primitive, something that's probably not even an animal at all. While some of the organisms in the Wang-En Biota might seem like animals, none of them actually were. They weren't crown group animals, or even stem group animals. They seem to be some even older opisthecont branch that was destroyed by competition with the new creatures that emerged in the Cambrian explosion 541 million years ago. The fossils in the Doshanto Formation of south-central China detail the development of the earliest complex marine ecosystems. Dominating the seafloor are these thick microbial mats, these dense layers of slimy goo filled with layers of bacteria and larger organisms. They were surprisingly stiff, kind of like a layer of sticky biological mud. These dominated the seafloor ecosystem basically forever, but in several regions within the youngest layers of the formation, the bacterial mats were being disrupted by the emergence of larger sessile organisms towering above them, and tiny cryptic millimeter-scale organisms that were apparently capable of movement. They could dig their way through the mats, plowing through the heavy biological mud and leaving behind trackways, tunnels, and burrows that reveal their presence. This new ecological activity was disrupting these billion-year-old microbial mat ecosystems and transforming the entire seafloor habitat. Above these microbial mats, swimming in the water column, are a wide diversity of algae and cyanobacteria, the primary producers that build raw biomass for the rest of the food web. Primordial groups of plankton were developing from populations of ciliates and other such free-swimming microbes, and both the plants and the fungi were actively experimenting with more complex multicellular forms. The animals, the true animals, appear to have emerged at some point within the time window of the Doshanto Formation, as they had developed complex forms like the sponges of the phylum Porifera, which filter-fed on all of the organic particulates in the water currents, and the Cnidarians, which had a more carnivorous or predatory inclination, and would have sought out the beefier, free-swimming microbes in the water column. One thing that's notably missing from the Doshanto Formation are bilaterians. To see fossils of the first Ediacaran bilaterians, we must leave the Doshanto carbonates and black shales of south-central China and head afield to other places in the world to find other rock beds that hold fossils from the same primordial time period. For example, on the other side of the planet, there are two important fossil beds on either side of the North Atlantic. One of them is the Charnwood Forest in central England, and the other is the Avalon Peninsula in Newfoundland in eastern Canada. Let's begin with the fossil sites in eastern Canada, along the southeastern corner of the island of Newfoundland. In this southeastern corner, there's a thin spit of land, no more than 5 kilometers wide, that connects at the hip to a roughly H-shaped region called the Avalon Peninsula. In this rugged, fog-shrouded place, there are two large exposed strips of ash-formed sedimentary rock called Lagerstätten that originated in the Ediacaran period. I've talked about this material before, but this Lagerstätten is an incredibly effective material for preserving fossils, especially soft tissues. Some of the most remarkable and informative fossil discoveries have come from Lagerstätten deposits. One strip of Lagerstätten lies on the eastern coastline of the peninsula, overlooking the Atlantic Ocean. The other strip cuts between the two vertical lines in the H shape, a bit closer to the main island landmass. Collectively, all of these Lagerstätten deposits are known as the Mistaken Point Formation, which has been dated to between 578 and 560 million years ago. This means that it's slightly younger than the Doshanto Formation, and it provides an excellent continuation of the evolutionary history. At the southernmost tip of the peninsula, there's a 5.7 square kilometer UNESCO heritage site called Mistaken Point Ecological Reserve, where one can find the richest and most diverse fossil beds in the entire region. These beds are so rich, and the fossils so stunning and surprisingly complex, with relatively few simple species rapidly transforming into many more complex species, that this time period, documented by this fossil bed, became known as the Avalon Explosion. It was kind of like a more primitive precursor to the Cambrian explosion, where new body forms were tried out and tested, and those that were suitable for survival persisted and experienced further diversification. There have been a ton of really wild fossils extracted from the Avalon deposits, including creatures shaped like leaves or paddles, like columns, spindles, or cones, or highly branching and tree-like forms, and flat body types that radiate outwards from a central point or a central line. One of the more curious fossils in Avalon is the Fractifusus misrae, which looks sort of like a fleshy upturned tiger cowrie shell. It has a body form like an elongated football lying horizontally along the seafloor, with a zigzagging crease running lengthwise down the middle. Folded lobes or segments of tissue grow outward from this crease, curling downward around the body shape, giving this entire football body structure a wrinkled, striated look. The only evidence we have of their reproduction is the presence of propagule-like structures, sort of like the plants that produce stolons to create new nodes or pseudo-individuals across geographic space. These organisms send out stolon analogues and grow a new individual some short distance away, like a very specialized form of budding using a weird aquatic primordial umbilical cord. These fractifusus organisms belong to a diverse group called the rangiomorphs, along with the closely related arboriomorphs and frondomorphs, which all have this curious, elongated physiology with a central zigzagging line, with lobes of tissue growing out laterally in this alternating stepwise form. Their bodies are shaped like oar paddles, or like particularly thick and fleshy fern fronds. Many rangiomorphs have hard bodies with mineralized skeletons. They don't have muscles or joints as far as we can tell, but rather the skeleton was like what we see in corals or sponges, where the body form is simply reinforced and made resistant to compression and deformation. Okay, so that was a rundown on the Avalon Peninsula, but before I get too far ahead of myself, let's look at the other post-Docento Formation fossil bed that I mentioned earlier. In central England, there's a roughly circular region about 170 square kilometers called the Charnwood Forest. Most of it is rugged hills and grassy fields, studded with huge rocks left over from volcanic activity and geological deformation going as far back as 600 million years. The actual forest takes the form of thickets and stands of trees scattered in the ridges and valleys of this rugged landscape. It's really scenic and beautiful, and an extremely scientifically valuable area. In the heart of Charnwood Forest, there's a host of sedimentary rocks formed from volcanic debris flowing into a body of water. Not so much magma, but more like ash and sediment and other materials in the pyroclastic flow. In our current time period of interest, about 570 to 550 million years ago, this Charnwood area was submerged. Unlike the Docento Formation, Charnwood was not a shallow water environment. This area was deep under the surface, so deep that light couldn't make it all the way through, and the seafloor was shrouded in total darkness. We know this because when sediment is deposited similarly in shallow water environments, the material takes on particular forms and features that aren't seen anywhere in Charnwood. The rocks themselves have also been dated really accurately, thanks to an abundance of uranium-lead zircon crystals that can be found throughout the sedimentary column. Within these Charnwood formations, there's a rich abundance of macro-scale fossils. In fact, the first Ediacaran macrofossils found anywhere in the world were found here, in Charnwood. The first to be discovered were members of the Charnia genus, which appeared to be immobile filter-feeding animals, perhaps similar to, but more primitive and different in form than the sponges, although it's quite likely that they're not even animals at all, but instead belong to some even more ancient branch on the tree of life that's totally unknown to us. I think one of the more recent hypotheses is that they belong to a large but now extinct branch, perhaps originating from Opisthokonta, closely related to the presumably older fungi lineage and the younger lineage of true animals. The carnia are shaped like a thin leaf, or a frond, or a tongue or a paddle, sticking tens of centimeters tall out of the ground from a single central anchor. The upper surface of this anchor sprouted 2 lobes, which parted to the side to make room for more lobes, budding out of the central proto-meristem analog. This bud would initially fall to the left or right side, and every subsequent lobe would grow out to the opposite side from the lobe that preceded it. In other words, the nodes didn't bud out in mirrored pairs, as they often do in plants, they budded out in alternation. This process, repeated, would create an extended mass of tissue, of alternating lobes extending to form a zigzagging line running down the center, or the analogous pseudospine of the organism. Most interestingly, the body plan represents the simplest kind of fractal pattern. Pairs of branching lobes, each lined with small segments or lobes of its own, which were themselves lined with even smaller protrusions, and so on and so forth. this is a kind of idealized fractal form that could be established with a relatively simple set of genes. And the more I think about it, the more it blows my mind. When we look at these organisms, we're seeing some of the first multicellular biochemical superstructures, and their physical structure unambiguously follows a mathematical format of a simple structure being repeated fractally as much as possible. When I look at this, I see the same kind of universal mathematics behind the ripples in a pond disturbed by a thrown rock, or the pressure waves of sound moving through a medium. These organisms are shaped by the exact same mathematical, physical, universal forces, making them kind of like the simplest version of a waveform, manifest not as ripples in a medium, but as the morphology of a living carbon-based substrate. It is the sublime beauty of the universe being clearly expressed in the shape of the first generation of mesoscale animal or animal-like organisms. And that's awesome! Anyway, the Churnia is a creature shrouded in mystery. There's a lot of speculation about all aspects of its life, but we don't know for certain how it fed or how it reproduced or how it avoided predators, if there even were animal predators around at the time, which honestly there probably weren't quite yet. Very little is concretely known about the Churnia, and almost everything is heavily speculative to one degree or another. For example, there's strong evidence to suggest that they lived in pitch darkness on a particularly deep water patch of seafloor, which has naturally led to the hypothesis that they were filter feeders, although the specific mechanism by which this happened is a mystery. It seems most likely that their bodies formed a simple corrugated paddle, or a frond, that could catch organic particles in the water current, which would then be pushed or filtered into the crevices between the lobes, where they would perhaps be absorbed. Or, alternatively, perhaps they were in the early stages of evolving more rippled or wavy lobes with more surface area, allowing them to more effectively absorb not just the tiny detritus, but the actual, dissolved organic carbon in the water. The fossils in the Avalon Peninsula cover a relatively brief time period, about 578 to 560 million years ago, whereas the Charnwood area has numerous fossil beds that cover a much broader range of geologic time, almost 10 times as long, actually, with sedimentary rock deposits from 600 to 450 million years ago. The amazing part is that when we use really accurate dating methods, like measuring the ratio of lead in a uranium-rich zircon mineral, we can line up the fossil beds from these sites in chronological order and compare the resident fossilized organisms to see how the global biota, or at least this Atlantic biota, changed over time. Of course, scientists have done this, and they found that the same types of organisms existed in both areas around the same time periods, as these two locales are relatively geographically close, and they would have been deep-water seafloor habitats with similar environmental and ecological conditions. Rather surprisingly, in fact, all of the organisms found throughout the world during this whole time period, from 578 to 560 million years ago, so this smaller period of time bounded by the fossils in the Avalon Peninsula, all of the organisms that existed throughout the world at this time are collectively known as the Avalon Biota, in honor of the discovery of the fossils in the Avalon Peninsula. Since these first discoveries of Ediacaran fossils in the mid-20th century, at least 25 more sites have been discovered all over the planet corresponding to this time period. They include fossil beds found in the remotest stretches of Siberia, to those baking in the heat of the Australian outback, to the many fossil sites across North and South America, and many more in China and East Asia. When all of these fossil beds, all of these excavation sites, are time-calibrated with those particularly useful uranium-lead zircon crystals, we see the same types of organisms fall into place in the same time periods all around the world. Studying these sites and their chronological order has massively informed our effort to understand the living world as it was more than 500 million years ago. For example, we now understand the Ediacaran Biota as having existed in 3 major stages, or assemblages, named after the important fossil beds that document each time period. The Avalon assemblage, which represents species that lived between 578 and 560 million years ago, is followed in turn by the White Sea assemblage, from the White Sea fossil beds in Russia, with species that existed between 560 and 551 million years ago. And this is followed by the most recent time period, the Nama assemblage, from 551 to 538 million years ago, and this takes us right up into the Cambrian explosion. As we move forward with our exploration, I'll be referring to these Ediacaran organisms according to the assemblage, or the biota that they belong to, and not necessarily by the specific site where they were found. This will just be a bit easier for everybody in general, and not just for the fact that there's many species that are found in multiple fossil sites. There's also a really important point that I need to clarify. The names of these time periods are just a useful convention. They don't actually strictly correlate to the specific fossil beds in question. For example, in the Avalon Peninsula, the types of organisms we see here are found elsewhere in the world, over a much broader time period than what we just see in the Avalon site itself. An even better example is the White Sands area in Russia, which is known to have fossil beds containing all 3 biota types in close proximity. The Nama assemblage, which formally covers the last few million years of the Ediacaran, saw the emergence of new lineages that would survive the Cambrian explosion and outlast the rest of the Ediacaran biota by tens, or in some cases even hundreds of millions of years. So the practical import of this, as far as we're concerned for our narrative here and for understanding the evolutionary history of the Ediacaran, is that all of these different biota types, the actual animals that are represented in the Avalon, in the White Sea, and the Nama assemblages, they likely coexisted in time. They simply represented different marine habitats, different groups of organisms in distinct environments at different latitudes and at different depths in the ocean, but with many species showing considerable overlap in geographic and temporal space. Now, with this said, we will also explore some of the many macro trends that were affecting entire communities and populations and ecosystems over the many millions of years of the Ediacaran. So we have a lot of things going on at different rates. Some groups are evolving and diversifying at a certain rate, other groups are dying off, perhaps at a slower rate, and it's all scattered piecemeal in dozens of different fossil sites scattered around the planet. This is a complicated 4-dimensional puzzle, not just with X, Y, Z geographical components, but also with a time component as well. Let me give you a couple examples of this nuance to try to simplify everything and illustrate the issues that I'm talking about here. Perhaps the most obvious example is the name of the Ediacaran period itself, which comes from the Ediacaran Hills in the Flinders Ranges of South Australia. Now, these fossil beds in the Flinders Ranges only cover a small fraction of the total Ediacaran period, and yet the naming convention is what it is. Another example is the Ventogyrus. These were really weird animals that existed at least 635 million years ago, way before the Avalon time period, and they thrived in the White Sea time period, and only disappeared at the very end of the Nama time period, 542 to 540 million years ago, give or take. Now, despite existing throughout all 3 of the major divisions of the late Ediacaran, they're grouped with the White Sea biota, because the White Sea fossil beds in western Russia, near Finland, are where we see the greatest abundance of these Ventogyrus creatures. These details about the Avalon, the White Sea, and the Nama assemblages, and these naming conventions, are really important bits of nuance that you should keep in mind as we move forward. But briefly, before we really get started, I wanted to mention that the word Ediacaran itself has an elusive origin in the languages of the Australian indigenous people. The actual origin of the word is unknown, but a popular idea is that it's a mispronunciation of the term Ediacaran, meaning hard or rocky ground, or granite plain, after the large shelves and plateaus of dolomite rock that exist in the area. I just thought that was interesting and worth mentioning. Getting back to it, let's start with the earliest of these 3 late Ediacaran time periods, divided as they are into the biota types. This first time period is characterized by the Avalon biota. This, of course, includes the rangeomorphs, with the 2 examples we looked at a moment ago, from Charnwood in England and the Avalon Peninsula in eastern Canada, and these cryptic organisms were sessile filter feeders. Many species grew on a vertical axis up from the seafloor, like Charnia, while others grew horizontally along the seafloor, like Fractifusus. Both of them had fractal-like bodies, structured like soft, fleshy fronds, analogous to the leaves of a fern. The Charnia was like an upright fern, or like a plant standing up in the sunlight, whereas the Fractifusus was kind of like if you took the fern and wrapped it around a football. That's sort of, roughly what it looked like. But other rangeomorphs showed greater variation on this theme, with some species having long stalks between the frond portion and the anchor tissue holding it to the seafloor. Both of them had fractal-like bodies, structured like soft, fleshy fronds, analogous to the leaves of a fern. Charnia, for example, stood upright like a typical plant, turgid in the sunlight, while the Fractifusus was more like if you were to wrap this fern frond around a football. Sort of. Other rangeomorphs showed greater variation on this theme, with some species having long stalks between the frond portion and the anchor tissue holding it to the seafloor. This is reminiscent of crinoids, a relative to the starfish, although those are much, much more complex than anything we see here in the Avalon biota at the time. There were some groups, like the Frondophilus genus from the early Avalon assemblage, that had a surprisingly long central stalk. One fossil specimen was a meter long, which would mean it was one of the tallest organisms around in the Avalon biota. It would have towered over the bacterial mats and the smaller rangeomorphs, and the new corals and ancestral seaweeds. Unusually, the Frondophilus possessed lobes that were not arranged in a continuous feather-like or frond-like pattern, but rather they had discrete leaflets, little fractal lobes that branched out of the main stem at equidistant points such that they didn't overlap one another. This may have been an adaptation seeking to increase the surface area or receptive field that could catch food particles from the water current. It would have also avoided any potential issues with abrasion or scraping and damaging of the epidermal tissue from having the fronds rubbing against each other in the current. As the Avalon biota time period came to an end around 560 million years ago, we started to see a stark increase in biodiversity and the complexity of body forms, and this continued and accelerated into the White Sea time period, which spans about 560 to 551 million years ago. The name of this time period and the associated fossil assemblage comes from the fossil beds near the White Sea, a hook-shaped body of water looping around the underside of the Kola Peninsula, in the northwestern corner of Russia. Along the southeastern coastal areas of the White Sea, there's a region that's mountainous and rugged. It's a picturesque rocky alpine area in the summer, but in the winter, it's blanketed in thick sheets of snow. Within these rugged hills, there are layers or lenses of sedimentary rock rich with fossils, with the two largest and richest of them being near Zimny Gori and the other near Solza in the Arkhangelsk region. Both fossil beds were originally a shallow water ecosystem in a basin along the sloping edge of the continental shelf. regular rainfall and snowmelt eroded the more rugged dryland areas, and a gradual influx of sediment was normal, but occasionally the erosion would prove destabilizing, and a great mudslide or rockslide type event, perhaps in combination with an avalanche, carried material far out across the continental shelf. So imagine, if you will, a whole bunch of sediments falling loose in a mudslide and cascading down a mountain, plowing into a shallow water basin, a little shallow coastal area there on the continental shelf. This event buried all of the organisms in this shallow water basin habitat, and it preserved them in extremely high detail. It's really interesting to note the major ecological and physiological shifts seen across the Avalon, the White Sea, and the Nama assemblages. In particular, a pattern of increasing complexity from the Avalon to the White Sea time periods, followed by a struggle to maintain that complexity and biodiversity in the later Nama time period. For example, in the Avalon, the biota is virtually all found in deep water environments along the remote seafloor or the most distant slopes of continental shelves. In the White Sea and the later Nama assemblages, we see organisms having spread out to a wider variety of marine habitats, including the middle and inner shelf zones, and even the shallow water areas near the coastline. Virtually all of the Avalon biota was fossilized through siliciclastic preservation modes, like with silica-rich volcanic ash and silicates from rocks that were finely ground down by Ice Age glaciers, and later, when the glaciers receded, they were washed into the sea by the rain. In the White Sea assemblage, about 1 in every 6 or 7 fossils were preserved via carbonaceous compression, which is much more common in shallower water environments. And in the Nama assemblage, more than 3 out of 5 fossils are preserved through the older siliciclastic processes, with the rest, about 2 out of 5, being preserved through, in increasing frequency, carbonaceous compression, carbonate mineralization, and pyrite replacement. All of these patterns in mineral fossilization indicate a shift to shallower water habitats, shallower water environments, where minerals like carbonates and the materials necessary to create them were much more common. Another curious pattern is the prevalence of feeding strategies and life habit. In the Avalon, there were really only 2 feeding modes. You had osmotrophic absorption of dissolved particles for nutrients, typically based purely on diffusion, and you had suspension feeding, which involved creating broad surfaces to catch and absorb or ingest larger particles, like decaying detritus that had been swept up in the current or something. But in the later White Sea and Nama assemblages, organisms that use osmotrophic feeding strategies have plummeted in abundance. They're being rapidly outcompeted by organisms with more efficient feeding strategies, like the suspension feeders, and in the White Sea period, there were also emergent predators, and the mat feeders like Dickinsonia and others, who roamed across the seafloor absorbing through their belly the nutrients in the bacterial mat. Other types of surface-crawling organisms emerged too, that scavenged for food on the detritus-rich sediment. However, in the Nama time period, it seems like populations were decreasing across the board, and detritus was becoming less common, as the few scavengers there were in the White Sea time period seemed to have disappeared in the Nama. Similarly, the bacterial mats were heavily degraded during the White Sands time period, and by the Nama time period, they weren't able to support the populations they once could, so the abundance of mat-feeding animals, or mat-feeding organisms, was pretty much cut in half. A similar pattern can be seen in the life habit, or the general mode of existence, of the Ediacaran Biota. In the Avalon, almost everything was immobile, or sessile, and firmly attached to the substrate. A bit fewer than 10% of species were sessile, but not firmly attached to the seafloor, and they could be rolled around by a sufficiently strong current. organisms capable of autonomous movement, like crawling across the seafloor, or even swimming in open water, and possibly even predators, appeared in the White Sea time period. Although, when conditions became more challenging in the Nama, these free-moving organisms were drastically reduced in abundance. Okay, so let's get back to some of the actual White Sea biota. I mentioned that there was a surge in biodiversity going into the White Sea time period, and this includes new lineages that would die out, and some that would live on and diversify into the modern day. For example, consider the Cnidarians, like the sea anemones, and the jellyfish, and the siphonophores. Many primitive Cnidarian forms have been found in the White Sea fossil beds near the Kola Peninsula, like the Bonata septata, a 4 centimeter wide, disc-like creature with a central visceral mass, shaped sort of like a flying saucer. A similarly shaped 6 centimeter wide species is known only by one specimen, called Veprina, which is thought to have been an active seafloor roaming predator equipped with tentacles that preyed on the more herbivorous grazers. And there's also the Inaria, an apparent filter-feeding organism shaped like a hollow clove of garlic, or a pumpkin, with a thick, chimney-like stem growing out from the top. Other weird creatures in the White Sea fossil beds include the Caretza brutoni, a small, bilaterally symmetrical trilobite-looking animal with multiple antennae growing out of its face. The cryptic Margarita flabellum, a fan-shaped grazing organism that eludes classification. And there was also the Zolotitsia, a strange, sessile animal, probably an ancestral Cnidarian offshoot, that grew like an old rangiomorph. That is, it looked like a long leaf with a central stem anchored to the seafloor. Except instead of 2 halves of a green leaf extending out from the stem, it had a series of oval-shaped segments, or organs, organized in rows running down either side of the stem. Perhaps it was a filter feeder that caught food in the creases of its oval-shaped segments, but honestly, who knows? It's a particularly strange and unusual animal, like so many other members of the Ediacaran Biota. One of the potentially more important fossils in the White Sea biota, seen not just in the White Sea fossil bed in Russia but all over the world, is Kimberella, a soft-bodied, oval-shaped, slug-like creature with a stubby, thumb-like protrusion for a head. They were smooth and rubbery creatures, with a protective, fleshy pad along their back, akin to the stiff but non-mineralized shell of a leatherback turtle. The body was unsegmented, but it had rings and lateral strips of muscle that allowed it to stretch their body to twice its normal length, and wiggle the evolving foot organ to move across the seafloor. This flexibility seems surprising in an animal that also has kind of a stiff shell, but it appears that the shell wasn't a single cohesive sheet of tissue, like a toenail or a monoplacophoran. Rather, it seems like their shells were kind of like connective tissue, an aggregation of cells and particles in a flexible matrix. In this case, Kimberella is thought to have had an aggregation of mineralized spicules, like what we see in the sponges, except much more densely arranged in a much thicker collagen fiber matrix. This type of a structure could account for the flexibility that we see in the specimens. Now, I just mentioned the monoplacophorans. These are really primitive mollusks characterized by an oval body shape, with a central mineralized shell plate on the back, and a rim or an apron of soft tissue extending out around the shell plate. This apron of tissue contains sensory organs and muscles to help it move and swim. Kimberella also exhibits a rim or an apron of soft tissue that extends out beyond their soft shell dorsal covering. This blanket of tissue has rippling, wavy edging, a fringe with a high surface area to volume ratio, which is thought to have been used like a primitive set of gills. The fringe is actually quite large, which has led to speculation that Either the absorption mechanism was inefficient, and more tissue was needed to compensate, or there simply weren't any major predators that lived at the time and/or the place that Cambriella lived, so these extraneous and easily ensnared frills weren't necessarily a safety hazard. The fossils of Cambriella have been found in areas that were once shallow water habitats with exposure to sunlight. Here they would have been more exposed to the atmosphere and the terrestrial world, so things like landslides and mudslides, or heavy rains and storms that created strong waves, would all stir up sediment in this shallow water habitat. Cambriella would have had to deal with these occasional aquatic sandstorms, or the rapid deposition of sediments, which may have been the evolutionary impetus to develop muscles that could retract the head and the tail under the protective shell. If they got buried, they would try to push upwards to reach the surface again. And there's even fossils of individual Cambriella at the top of small vertical tunnels, suggesting that they tried to dig themselves out of the mud, but they ran out of energy and died. It's likely that Cambriella was a grazing animal that fed on bacteria and algae in the microbial mat. It would have slimed its way along like a slug or a snail, pushing through this microbial mat and absorbing nutrients. Distinctive scratch marks have been found on the rocks near Cambriella fossils, specifically in lines radiating outwards from the head of the nearby fossils, which led to a hypothesis that they were made by a radula, a hard, tooth-like mouthpart seen in mollusks. The idea is that the individual Cambriella would position itself in a suitable spot, and then it would stretch out its neck to scrape its radula teeth, or tooth, against the rock. This would scrape off algae and bacterial slime, which could then be gobbled up and ingested. Like a massive earth-digging machine swiveling its arm around to clear out a semicircle of dirt, Cambriella would swivel its extended head to graze on a semicircle of bacteria and algae before gliding over the harvested area to move to a new spot. A similar feeding mechanism can be seen in sauropod dinosaurs, who used their extraordinarily long necks to graze on foliage in a massive semicircular area without having to move their body too much. Cambriella was known to exist from at least 558 to 555 million years ago, as determined by the thoroughly well-dated rocks in the White Sea area in Russia. Cambriella fossils can be found outside of the White Sea area as well, like in the Ediacaran Hills in South Australia, which have fossil beds known to be both older and younger than the White Sea fossil beds. So it's quite likely that Cambriella existed in a slightly broader window of time than what we see in just the White Sea deposits. Now, if we combine all of these things, this mollusk-like body plan, the mollusk-like feeding style, and the date of its emergence, things get really exciting and more than a little bit controversial. Although Cambriella was initially mistaken as a jellyfish, new fossil discoveries have led to wide agreement that Cambriella is, indeed, a bilaterian triploblast, demonstrating clearly that bilaterian animals with complex and highly differentiated tissue had, in fact, evolved by this point and produced a healthy degree of biodiversity. But controversy sets in when trying to figure out what kind of bilaterian it is, or pretty much anything else about it. It's probably a protostome, and not a deuterostome, due to it being an invertebrate and having multiple similarities with mollusks. But we don't actually know what direction it moved in, with some thinking it moved backwards and grazed on bacteria and algae that it had already passed over, while others think that it moved forward and grazed on what was in front of it. Furthermore, the scratch marks from this grazing behavior from this supposed radula feature are the focus of a really heated debate. Arguments have been put forward that the scratch marks are caused by a pulling motion, where the head is first extended, the radula then placed on target, and then the head retracts, scraping the teeth along the ground to sweep the loosened food particles towards the mouth. This is not how modern mollusks do it, as they push their radula forward, like a chisel being pushed through wood or a shovel biting into the dirt. The controversy compounds when you learn that there aren't actually any examples of fossilized Cambriella radulas. We don't know for certain if they even had them in the first place. It's purely speculative based on a lot of this circumstantial evidence, and the scratch marks themselves are merely another bit of circumstantial evidence. While the radula aren't known to readily fossilize in actual unambiguous mollusks, many see the total absence of radula fossils in or near Cambriella specimens to be a fatal strike against the hypothesis that they're ancestral mollusks. At most, a tentative case could be made that they're an offshoot branch of a stem group protomollusk that existed long before the crown group mollusks emerged. Depending on who you ask, even this is in debate, and they argue that we might be going too far by even calling them bilaterians. So Cambriella remains one of the more curious and controversial mysteries of the Ediacaran Biota. Another notable group that emerged around this time was the Trilobizoa, which existed from 558 to 550 million years ago. These are particularly strange animals because, while they were relatively flat, disc or mound-shaped organisms that lived immobile on the seafloor, all common qualities of ground-based grazers and scavengers at the time, but they display a rare and unusual tertiary or triradial symmetry. All of the Trilobizoa are flat, roughly circular, disc-shaped organisms. Within the structure of this body design, the tertiary symmetry manifests itself in several ways, depending on the species. The most characteristic genus is Typrachidium, which are usually no more than 4 centimeters wide. Now imagine 3 fat, wrinkly slugs swirling around each other, nestling their heads together to make a twisting gumdrop-like body form. This is sort of what the Typrachidium looks like. It's really strange, and unlike almost anything else we've seen. Trilobizoans appear to be immobile. Most groups are flat discs, maybe slightly domed, with 3 small dimples or depressions in the center. There are ruts or channels that run from the central depressions to the outer rim, and these are believed to be part of a digestive system for circulating nutrients around the body. Some Trilobizoans, like the rugoconites, are found in clusters, which has led scientists to think that they might exhibit a form of social behavior, or at least communal living. Other groups, like the albumaries, have 3 rounded leaf-like lobes. The Infesta have 3 fat, finger-like ridges spiraling from the center to the outer rim, each ridge rotated 120 degrees from the others. Another triradially symmetrical creature is the Coronaculina acula, although this doesn't seem to be a member of the Trilobizoa. The Coronaculina acula is a bulbous, cone-shaped creature whose base sits on the seafloor, and whose tip is opened up like a little drawstring bag. This cone shape is subtly divided into 3 lobes, tapering to 3 little bumps along the ring-shaped opening at the top. Perhaps most strange are its 4 spicules, which are a rare example of mineralization in the Ediacaran Biota. These spicules take the form of long, narrow, pencil-like spikes, which grow from the upper part of the body near the open rim, but lean outward against the seafloor, making a plus sign against the ground. Coronaculina acula has only been found in a fossil bed in South Australia dating to 555 million years ago, which means that it could be an Ediacaran relative of the Koya, a type of demosponge that appeared in the Cambrian, which also possesses these long spicules radiating outward from a central cone-like body mass. Another strange, radially symmetrical animal is the Arkarua adami, with specimens found in the Flinders Range in South Australia, dated to about 555 million years ago. These look like sand dollars, in the sense that they're small, flat, disc-like organisms with a unique 5-point radial symmetry. There are 5 equidistantly spaced depressions on their upper surface. They're speculated to be ancient echinoderms, or basically an ancestral or really basal stem group offshoot of the starfish. However, only their external features have been preserved in rock, so we can't see their inner organs. And they also don't seem to have stereome plates, which are features seen in all modern echinoderms. So, while we suspect that they might be primitive echinoderms, it's really just a working hypothesis. Their actual affinity hasn't been conclusively determined. I should mention that the Flinders Range in South Australia is a rich fossil bed, dated to about 555 million years ago, with a great density and diversity of species. I've already mentioned a number of these, like Arkarua adami, Coronacolina acula, and a number of other trilobizoans. But this is just a small sample of all of the weird creatures fossilized in the sediments of this Adelaide superbasin. Some other strange creatures that you'd find in these Ediacaran seas include the Paleophragmedictia, an extremely primitive and early sponge, perhaps a glass sponge, maybe, from 558 to 555 million years ago. The Paleophragmedictia was shaped like a puffy dome, with 6-fold symmetry in its ridges and long spicules radiating outward. Some species had a tentacle or a stalk, or some sort of long appendage that is thought to have extended into the water as some sort of filter-feeding structure. Another species found here in the Flinders Range is the Ovatoscutum concentricum. It's a flat, round, Pac-Man-shaped creature made of a large arcing segment, or a lip, that runs around the circumference of the creature's body. A Z-shaped or zigzag line runs down the middle, created by the alternating internal segments, or the ribs, that fill out the rest of the circular or Pac-Man body shape, with what looks like concentric rings. Take note that the head would have been at the center of the rounded edge segment, with the Pac-Man face-looking side, or if it was like a pizza with a slice taken out, the space where that slice used to be, that part of the body would be the tail region, or the back side. These Ovatoscutum concentricum are seen in contact with the microbial mats, suggesting that they too roamed the seafloor and absorbed nutrients directly from the slimy, muddy microbial mat. There's a somewhat controversial group called the Hymalura, which emerged in the Avalon but existed throughout the entire White Sea period and disappeared at the start of the Nama. The Hymalura appeared to be circular sea anemone-like animals with no hard parts. But this interpretation is controversial, because a lot of the purported Hymalura fossils have turned out to be something else, something like the anchors or the holdfasts of larger vertically oriented filter-feeding animals like the Primocandelabrum, which are really similar and closely related to the Hyliculus from the Avalon, those unique filter feeders whose lobes reach upwards to form a cup or a goblet shape. Another strange group in the White Sea biota, dated to around 555 million years ago, are the creatures of the Pambacalbi genus. Like many of the other rangiomorphs, these had an anchor mass, a central stem or a column, and multiple veins lined with fractally branching appendages in a frond-like pattern. The Pambacalbi is known from a single species, with specimens found only in South Australian fossil sites. Despite their really tightly limited geographic and temporal distribution, they exhibit a remarkable feature that we can't ignore. Instead of fronds that branch off into smaller and smaller appendages in a simple fractal pattern, the Pambacalbi have fronds that form rows or clusters of small cups or bowls. These little hollow nodules have a small opening at their upper point, where water and nutrients can flow in and waste material can be expelled. It's believed that inside of these nodules, inside of these hollow bowl-like structures lining the veins of the Pambacalbi, were flagellated cells like choanocytes, whipping up the water currents and facilitating absorption of nutrients. Keep in mind, the presence of flagellated cells like this is a major link between the sponges and the closest non-animal microbial relative to the animals, the choanoflagellates. Thus, the Pambacalbi represents an incredible and important evolutionary achievement. It's not known for certain if these organisms are ancient cousins of the sponges, or like some weird stem group branch that's long extinct, but they clearly demonstrate another major breakthrough in the evolution of more complex body forms. To be a bit more specific, this breakthrough is the ability to filter feed in smaller, controlled microhabitats, which gives you a better chance of catching larger food particles and improves the overall efficiency of your filter feeding. In a more general sense, all of these rangiomorphs belong to a larger group called the Petalonymi, which are named after their flower petal or leaf-like body shape. I've been referring to the body shape of the rangiomorphs as a fern-like frond for pretty much the entire episode. There were also curious oddities at the edges of the Petalonymi, like the Stromativarus. This organism, much like the rangiomorphs, stood vertically from an anchoring point on the seafloor, with a frond-like structure waving in the water current. But the stalk connecting the anchor point to the frond was unusually thick, as if engorged with fluid or some sort of complex internal arrangement of tissues, like a coelom or operculum cavity, or even a group of rudimentary organs. The Stromativarus is suspected of being a particularly close relative to the true animals, if not an extremely basal animal in its own right. There's an idea that they could be immobile, ground-anchoring ancestors of ctenophores, of the comb jellies, although there's not enough data and analysis done yet to come to any definite conclusions in that regard. And then, of course, there were the true animals themselves, those organisms that we are confident are actually part of the clade Animalia, and not some mysterious, long-extinct branch from way earlier in the history of life. Among these Avalon animals, we've found fossils in Charnwood Forest of the earliest known animal predator, which dates to about 560 million years ago. This was the Aurora lumina, a genus of incredibly ancient anemone-like cnidarians. The genus is known from a single species, with a single specimen. Its body represents two bifurcating stalks, each ending in a wavy cup or bowl-like structure that houses a nest of stinging tentacles. This is yet another example of a new breakthrough adaptation appearing in the eukaryotes, as the evolution of stinging cells, or cnidocytes, was a major boon for catching prey. A similar organism, also found in the Avalon Peninsula, is the Hauschia quadriformis. This too is a primitive cnidarian with an anemone-like lifestyle. Anchored to the substrate by a stalk, the body of this polyp form opens up like a four-sided cup, about 6 by 4 centimeters in size. At each corner, extending loosely upward into the water column, are 4 branching tentacles, with a host of smaller tentacles protruding from the edges or the rim of the cup shape of the body. These tentacles were presumably equipped with primitive cnidocytes to stun prey animals passing nearby, paralyzing them so they can more easily be brought towards the mouth, ingested, and dissolved. These tentacles were presumably equipped with primitive cnidocytes to stun prey animals passing nearby, paralyzing them so they can more easily be captured, ingested, and dissolved. The Hauschia quadriformis is worth mentioning for another reason, aside from being another early, albeit immobile, predator. It's the first animal that shows unambiguous evidence of muscle tissue. large proto-muscle fibers wrap around the cup in horizontally arranged parallel rows. This stunningly sophisticated arrangement would have allowed the creature to perform several purpose-driven movements. Small contractions could shake the cup, causing the tentacles to wave around and cover more area in their blind, grasping search for prey. At each corner or edge of the cup where the muscle fibers connected, they taper together and they stream upwards into the tentacles. This suggests that the tentacles may have been able to retract, or roll up into a protective ball-like form, or perhaps they were dexterous to some degree, and they could, after capturing a prey animal, intentionally bring it closer to the central body. As the animals evolved, another major development would be segmentation. Segmentation was a biochemically efficient way to organize a body, and it provided new ways to move and articulate. The evolutionary development of segmentation was a long and complicated process. It didn't happen overnight. There's a group of Avalon animals that fills in some of the space between the arthropods and other segmented bilaterian animals and the more primitive, sessile, leaf-shaped filter feeders. This incredible group is known as the Proarticulata, with specimens extracted from rocks dated to between 567 and 550 million years ago, meaning they existed from the mid-Avalon time period through the entire White Sea time period. The classic example of the Proarticulata is a creature called Dickinsonia, which looks like a flat disc that Roombas around the seafloor, soaking up nutrients and whole bacteria from the microbial mat. They share a remarkable similarity to the rangiomorphs, except that they aren't sessile. Dickinsonia appears sort of like the frond structure, but lying flat on the ground, and able to squirm and wiggle itself around to move. It's sort of like the rangiomorphs, except instead of fronds, Dickinsonia has what are called isomers. Think of the isomers as long rows of segmented tissue, sort of like soft velvety pencils lined up side by side. Row upon row of isomer radiate outwards from a central line running down the length of the body. This central line is zigzagging, like what we see in the rangiomorphs. Isomer segments protrude out left and right in this alternating pattern, creating a body form that's almost bilaterally symmetrical, but rather it has what's called glide symmetry. This is where it's technically symmetrical if you flip it across its longitudinal axis and then slide it forward or backward a little bit to line up with the alternating isomers. The largest isomer is a broad segment of tissue situated right at the front of the organism, setting the stage for this primordial proto-head to accelerate down the evolutionary path of encephalization. At the other end, where the two halves of the organism meet together, the tissue isn't arranged in isomers, but is instead simply bunched up and wrinkled, like a wet accordion, to bridge the gap. These Dickinsonia possessed an effective, if somewhat asymmetrical, animal form. Their fossils are typically found at the end of trackways dug into the microbial mats, which is really strong evidence for their ability to move autonomously. Essentially, you can see the evolution of directional movement, which is integrated with the evolution of the head and the tail. We're seeing the bilaterian body plan start to take form in these flat, disc-like creatures that otherwise look like a circular arrangement of drinking straws, or a tiny living doormat that slides along the ground. Seeing the development of bilateral symmetry, with a front and back and left and right sides, in the fossil record, and knowing how these features would go on to be hyper-successful and become super common in the animal kingdom, it's just incredible. Being able to find the fossil evidence of this feature in its most ancestral, atavistic, experimental stage, it's just mind-blowing. Anyway, Dickinsonia is part of a larger group of similar organisms. There's the Vendiomorpha, which are thought to be related to or even descended from the Vendobionta, like Ventogyrus. They have a broad head segment and a small number of thick, semi-overlapping isomers reminiscent of a horseshoe crab. Then there's the Dipleurozoa, which includes Dickinsonia and other similar relatives, and the Cephalozoa, named after their grossly enlarged and pseudo-vascularized head structure. Both the Dipleurozoa and the Cephalozoa have a much higher number of isomers, but these are much narrower, like pencils or toothpicks arranged in an oval or a circular shape. Another Vendiomorpha group, called the Vendia, has intermediate-sized isomers. The cephalozoans are the most diverse group among all of these flat surface dwellers in the fossil record, including a variety of forms with distinct patterns to their isomers, like ridges or folds, or additional protective layers of soft tissue. For example, there's the Cephalonega, which are small at 6 to 7 millimeters wide, and the Tamga, which are 3 to 5 millimeters wide. And both of these groups are wide, disc-shaped critters that existed about 555 million years ago. They're both characterized by a cluster of isomers surrounded by a broad apron of unsegmented tissue, sort of like the placophorans. Another important group in the White Sea assemblage are the Sprigina and their suspected relatives, like Cyanornis. Sprigina is particularly well known because among all of the free-living surface-dwelling animals capable of movement, Sprigina is most morphologically similar to immediately recognizable animals like trilobites and millipedes and the like. However, despite its apparent similarity to some of these more modern groups, it's been surprisingly difficult to place Sprigina in relation to more modern animals. Comparisons to polychaete worms or basal arthropods usually start off strong, but then there's some lack of a critical feature or some other disqualifying detail, and so Sprigina remains in its own clade as a curious and cryptic representative at the base of the bilaterian animal lineage. Another basal bilaterian, similarly cryptic, was the Temnoxa molluscula, found in fossil beds dating from about 580 to 555 million years ago. These have a body that's 8 millimeters wide, with a shape similar to a long fingernail with a really puffy cuticle. The part that looks like a puffy cuticle is the head. It's an enlarged U-shaped structure that caps the anterior end of the body. It's believed that they would turn to face into the water current, and the large rounded head surface and the angular body helped water flow over them. Although there's no evidence of internal organs, it does have what appears to be a sticky foot pad and a linear, not radial, body pattern. This has led some researchers to suspect that it may be related to mollusks, although this is disputed, and its true placement is unknown. An even more interesting example is the Icaria wariutia, which existed from before 560 million years ago to their disappearance about 555 million years ago. These Icaria wariutia were small animals, no more than 1/3 of an inch long, shaped like rounded cylinders or like grains of rice, or like really tiny pieces of kettle corn, as some specimens appeared to be thicker on one end than on the other. All of these small, stubby, slug-like animals appeared to be bilaterally symmetrical, and the way they can be seen contorting or bulging their bodies has led researchers to suspect that they were like tiny, flexible capsules of smooth muscle, moving themselves along through the microbial mats and the seafloor sediment with a peristaltic action, similar to an annelid earthworm, or how your esophagus and intestines move ingested food. The most optimistic interpretations of Icaria wariutia suggest that it may be one of the earliest representatives of true bilaterian animals as we understand them today, with a coelom, a one-way digestive tract that goes through the body with a distinct mouth and anus, and a hydrostatic skeleton upon which is mounted lateral strips and circumferential rings of muscle. If this optimistic interpretation of Icaria wariutia is true, then this humble, grain-of-rice-looking late Ediacaran animal may be an ancestor or an extremely basal stem group relative of all modern bilaterian animals. If this is true, the implications are as beautiful and elegant as they are wild and incredible. A simple animal whose body was little more than a squirming muscular capsule, either Icaria or a close relative, would go on to diverge wildly and within 600 million years produce everything from octopus and dragonflies to snakes and eagles to whales and tigers and us, human beings, and all of the great apes, and all of the primates, and all of the closely related rodents and lagomorphs and everything else. All of these animals, pretty much all modern bilaterians, could very well have descended from Icaria wariutia or something like it. That possibility, and what it illustrates about the story of evolutionary history of life on Earth, of our branch of the tree of life here, is just indescribably beautiful and sublime. It's amazing. All right, so now in our journey through the Ediacaran Biota, it's about 550 million years ago. The White Sea time period has come to an end, and the third and final chapter, the Nama time period, is beginning. This time period will last from about 550 million years ago right up to the start of the Cambrian, about 542 million years ago. I mentioned earlier that the transition from the Avalon to the White Sea was characterized by a surging increase in biodiversity, in body forms and modes of life. But the transition from White Sea to Nama was more difficult, and saw life struggling to maintain the Ediacaran biodiversity that it had already achieved. This appears to be the result of a global anoxic event of some kind, although it's not yet been determined what might have caused this, or the full extent of the anoxic event, how bad it was, what caused it, what environments, what habitats it affected, and to what degree. Those kinds of questions remain largely unresolved. But we can see how much of the animal life at the time responded to these anoxic conditions. Those various species that were capable of movement were forced to settle in shallower areas, closer to the shore, where oxygenation from atmospheric exchange was still strong enough to keep them alive. In general, this was a time of great difficulty. While several new groups emerged, and many older clades were lost, including a few of the more familiar ones that have been with us for a while now, like the tall, feather-like Charnia, that iconic Charnwood forest specimen, as well as the shorter, aquatic bush or shrub-like Bradgashia, and the radially oriented Hymelora. These and many others were pushed out of the arena of life as the environment changed, or new forms, better adapted to the changing ecosystems, emerged and outcompeted them. Now, the Nama assemblage itself is named after the Nama group, a massive, sprawling geographic feature along the western coast of Namibia, which is a moderately large country immediately northwest of South Africa. In the Ediacaran period, this area was somewhat similar to the Doshanto region. It was a shallow water habitat, with a seafloor exposed to sunlight, and an influx of silica and carbonate-rich mineral sediment. These minerals were used as skeletal components in life, and as a substrate capable of high-quality fossilization in death. The fossils here were encased in sandstone, typically the result of a sand dune collapsing, or a mudslide flowing into the coastal water, or a storm or some other event whipping up sediment on the seafloor and depositing a layer of fine particulate sand on a bunch of organisms in the area. These hapless creatures would then be choked to death by the sediment. They'd be slowly mineralized and compressed over time into sandstone to forge high-quality, three-dimensional fossils. Alright, so to get us started, the Avalofractus is a new kind of mineralizing rangiomorph that shows up in the Nama time period, although these are much more geographically restricted to the eastern Atlantic, and they're quite small, with a 1 centimeter width and an average length of just 5 centimeters. It's been suggested that these organisms, just like many other rangiomorphs, reproduced asexually through fragmentation. Parts of their bodies simply broke off and then grew into new individuals. The stolon-like propagule method that I mentioned earlier is a more sophisticated form of fragmentation. Avalofractus appears to have used a more brutish and crude method. They grew continuously, but the body materials could only support so much mass, so every few centimeters of growth led to them likely cracking and fragmenting, leaving behind parents or adult Avalofractus specimens of a semi-consistent size. The broken-off shards and the fragments that cracked off of the adult Avalofractus would then go on to reconstitute themselves and grow into a new clonal individual. There's a few particularly tantalizing specimens whose taxonomic placement is as controversial as it is exciting. The Protechurus, for example, is a worm-like organism that was originally thought to be an extremely basal kind of echiurid spoonworm, or a priapulid penis worm, or a velvet worm, or even an ancestral arthropod. Like so many other members of the Ediacaran Biota, it's not conclusively determined what it could be, although there's a hypothesis that's growing in popularity that claims that these Protechurus guys are actually really ancient chordates, either precursors or really early offshoots of the animal lineages that would develop notochords and then spinal cords. Another controversial and exciting topic are the Ossia fenestrata and the Burkia hunti. The Ossia fenestrata is a super strange animal that looks kind of like a gourd. Imagine that you stuck the narrow end of the gourd in the mud, and the bigger bulbous end, sticking up into the water, was covered in longitudinal grooves and pores or dimples arranged in a regular grid-like pattern. At the top, they possessed a large orifice, an opening leading to a cylindrical internal chamber. Now, no one has any idea where to place this thing taxonomically. Some people think it's a member of its own unique clade that went extinct about 548 million years ago, while others argue that it might be an ancestral sponge, or like an older relative of the sponges. Perhaps the most compelling and eye-opening interpretation is that they are actually urochordates, really primitive chordate animals. This hypothesis suggests that the Ossia fenestrata is an ancient member of bilaterians that shows signs of developing a notochord. They've either diverged from the stem group that would give rise to the chordates, or they are actually a really ancestral chordate offshoot lineage. If this is the case, then their closest living relatives would probably be the tunicates, specifically the Acidiacea, or the sea squirts. These form clusters or colonies of simple, soft-bodied, transparent tubes that ingest food by bringing water in through a central orifice and filtering out food particles. The other species I mentioned, the Burkia hunti, is very similar and slightly older, appearing like a small, soft-bodied vase with latitudinal grooves and a similar set of pores or dimples arranged in the grooves. It has similar tracheal physiology to modern tunicates, and it's suspected of being either ancestral to them or some offshoot of the ancestor. Together, these are purportedly representatives of various groups ancestral in some regard to the modern tunicates. Another genus worthy of acknowledgement is the Nemopoikia, with relatively large fossils dated to about 549 million years ago. These were probably very basal sponges, or some sort of creature ancestral to the peripherans. Nemopoikia grew in a stepwise fashion, first colonizing an area with soft, fleshy tendrils, followed by a period of mineralization in which an aragonite skeleton was produced, and the body hardened. Individual organisms could grow up to a meter wide, as they tended to fill in crevices or gaps in the rock where they could safely filter feed. The Namapoikia is known to have coexisted with a few other interesting animals that also synthesized calcium carbonate skeletons, like the Cloudinidae and the Nemicolathus. Both of these groups were unusual, even among their contemporaries. The Nemicolathus, for example, grew as a thin stalk about 30 millimeters long, but it lacked fronds. Instead, it was equipped with a cup-shaped organ at the tip, or a spherical chamber with a hole on top, and 6 or 7 smaller pores running circumferentially around the middle. Depending on the species, the cup was a narrow little basin barely wider than the stalk itself, while others had an engorged sac-like cup organ that was almost as wide as the stalk is long. The stalk and the cup would both be hardened by a magnesium-enriched calcite skeleton that helped to keep it upright. Curiously, everywhere that we find Namicolathus, we also find Cloudinidae specimens, although the latter always seem to be less abundant than the former. The Cloudinidae are column-shaped organisms whose bodies are composed of repeating conical units nested within each other. I really didn't mean to use the ice cream cone metaphor so much, but if you think of like a stack of ice cream cones, like you'd see at any ice cream shop, that's what Cloudinidae looked like. The points of the cones aimed downward and were most likely embedded into the ground. This pattern of stacked cones formed a narrow column just a few millimeters tall, with one paper describing it as a funnel-in-funnel morphology. These small, hardened Cloudinidae are members of an informal collection of fossils called the small shelly fauna, or small shelly fossils. This really isn't the best name, as they're sometimes not small, and they're often not equipped with a hard shell, per se. They might have had just a simple hard part, like mineralized spicules, or an exoskeleton with a shape that no one would describe as a shell, like a branching forked shape, or in creatures where the living biomass is a thin sheet atop a hunk of deposited minerals, like a proto-sponge version of a bacterial colony's stromatolite. This group of small shelly fauna or small shelly fossils is also not a formal taxon or a formal clade in any sense. It's just a really general grouping of all of the small creatures, with bodies measured in millimeters to a few centimeters, who developed hard tissues like shells and spicules and whatnot in response to predation or environmental pressures. It wouldn't be inappropriate to make a comparison to the acritarchs, as the small shelly fauna also include detached shells, shell fragments, fossilized burrows left behind by shelled animals, and other various ichnofossils. It should come as no surprise, then, that the small shelly fauna group as a whole is extraordinarily diverse. A plurality of the group belong to these Cloudinidae things, these tiny columns of nested mineralized cones. But there's also the ancestral mollusks, which were small snail-like creatures with a hard shell. The ancestral brachiopods, which had evolved shell plates sort of like clams and oysters and other bivalves. And a swath of other creatures that did not survive for long after the end of the Ediacaran period. In the White Sea and the Nama time periods at the end of the Ediacaran, a new type of animal was emerging, one that would revolutionize the world's ecosystems forever after, and inflict such heavy evolutionary pressures on the other animals around them that they would be the driving force behind the evolution of shell plates, defensive spikes, protective behaviors, and all sorts of other defensive adaptations. This new type of animal was the predator, a carnivore that did not get its food through passive filter feeding, not through saprophytic or isotrophic absorption, not through photosynthesis, but through active heterotrophy. In other words, the predators revolutionized the food web because instead of competing with other organisms for food, like to get a good spot in the current to filter feed, or a good spot in the sunlight to photosynthesize, or a good spot on the microbial mat to graze on, instead of all of that, they would just hunt the other organisms around them, attacking them directly by tearing into their soft, defenseless bodies and consuming their visceral mass for its nutrients. I should clarify that heterotrophy technically already existed. Predation has been a thing for billions of years, as amoeba and fungus and other microscopic organisms consume bacteria and each other for food. This new kind of predation that I'm talking about is the kind seen in the macroscopic multicellular animals capable of free movement through the water or across the seafloor. It's likely that predation in at least some of these macroscopic animals is a derived form of scavenging. Those animals that earned their keep by scavenging detritus, like the biomass of a big dead animal, would find themselves adapting in multiple ways. Their digestive system would adapt to optimally digest the animal tissues, and their mouthparts would adapt specialized feeding structures, like sharp mineralized teeth or proboscis that can effectively damage the animal's body and extract flesh from it, so it can be consumed. When features like this had been developed, it really wasn't that big of a leap to go from eating dead flesh to eating living flesh. This would have been especially true when all of the potential prey animals around were soft-bodied, weak, and either slow-moving or totally immobile and defenseless. Other forms of predation, like that seen in the early Cnidarians, involved a specialized form of filter feeding, where the things being extracted from the water were no longer dissolved carbon or small organic particles, but rather macroscopic ediacaran sea creatures crawling or swimming around near the seafloor. This evolutionary shift was paired with the development of venoms that could harm or stun the prey animal and make it easier to capture and dissolve. The first generation of these mobile, active, intentional predators would have been unstoppable in this gentle environment, limited only by the rate at which they could move and or capture prey and the size of their gut. There was a new dynamic in the Earth's oceans, a new ecological axis upon which the animals could be divided, predator and prey. And while the existence of this new paradigm may have been an unpleasant realization for the prey animals, they quickly began evolving all manner of defenses against the predators. I've already discussed many groups with hardened exoskeletons. These would have initially given them structural support against the water current, first and foremost, but they would find new purpose in deterring and defending against the newly evolved predators. So, in the Nama time period, immediately before the Cambrian, we start to see signs of specialized tissues adapted for defense. The dorsal surfaces of many crawling organisms and the outer surfaces of many soft-bodied sessile filter feeders begin to integrate calcium compounds into their structure. These tissues were optimized into calcified exoskeletons and shells and armor plating that offered protection against the scraping prototeeth, the stabbing proboscis, and the stinging tentacles of the predators. We even see various types of algae begin to encrust their cell walls with a calcium-enriched matrix, literally like a cellular scale suit of biogenic armor against the emergent zooplankton predators. Some prey animals didn't adapt hardened, calcified tissues for protection, but instead, they changed their behaviors, they changed their life strategy and their lifestyles. They learned to burrow and tunnel into the sediment, to hide and avoid predators. Many of these animals went farther along this evolutionary pathway and became specialized for subterranean lifestyles. Some of them made extensive use of burrows and tunnels and largely retained their mobility, while others adapted to a more sessile lifestyle spent hiding right underneath the surface of the seafloor. For example, consider the Urnieta platoensis, which existed from 548 to 541 million years ago, as a member of the Nama biota near the end of the Ediacaran period. This animal was like an upside-down bell, partially buried in the mud and the sand in shallow water coastal habitats. The bell was composed of two shell plates, sort of like a bivalve, except instead of creating a sealed-off space, the fringes opened up outward to create an open space where water can move through and particles of food can be extracted. These Ernietta platoensis grew in clusters or bunches, and because of their shallow water habitat, it's suspected that they may have had a symbiosis with photosynthesizing algae. A competing idea is that they may have been filter feeders, who extracted their food with body parts made of soft tissues, perhaps akin to a sarlacc from Star Wars. This is only a hypothesis because, in the few specimens that have been discovered and described, the soft body parts simply didn't get preserved like the harder shell plates. For their part, the shell plates are composed of a sheet of small tubes arranged in parallel, with the tubes in each plate growing from a central seam or a fold. And this is where the similarity to bivalves becomes really apparent, but the seam itself is staggered or zigzagged. The tubes are not arranged in pairs on either side of the seam, but rather in an alternating interlocking pattern, much like the central lines seen on the older Ediacaran animals. Now, in response to the formation of harder tissues and burrowing behaviors, the predators developed adaptations of their own. I already mentioned the aurora lumina from the White Sea period. This is the oldest known animal predator, dated to 560 million years ago. It's a primitive sea anemone, meaning the cnidarians, and likely their cnidocytes, had evolved by this point in time. The cnidocytes were a remarkable and frightening adaptation. These are cells loaded up with toxic compounds, and a mechanical apparatus, kind of like a syringe or some sort of injector, that can deliver the venoms to the prey animal. Today, cnidarians have evolved a variety of neurotoxins that can inflict paralysis, pain, and physical damage to the prey animal. But in the Ediacaran, the cnidarian's retinue of venoms was almost certainly less diverse and less sophisticated. But presumably, it had some limited capacity to disable or harm the prey animal, and make it easier to capture and consume. Predators also evolved methods to break through or bore into the hardened tissues of their prey. For example, consider the Claudinidae again. These were the first members of the small shelly fauna that I introduced, that look like a tower of stacked ice cream cones. They had moderately to fully mineralized calcium carbonate skeletons, and in many of their fossils, up to 20% of them in some fossil beds, we can see evidence of predation, of damage inflicted on the skeleton, like scrapes and fractures, and most interestingly, bore holes. These fossilized injuries show that by the late White Sea period, and definitely by the Nama period, the predators had developed means of boring or drilling through hardened skeletal tissue. We're not even at the Cambrian yet, and we're already seeing the back-and-forth struggle of a so-called evolutionary arms race between predators and prey. The discovery of the boreholes in the Claudinidae fossils is pretty incredible all on its own, but it gets better. The size of the boreholes generally corresponds to the size of the Claudinidae specimen. Or to phrase this another way, the predators that could bore holes through calcium carbonate exoskeletons, they were seeking out prey animals that were appropriate targets for their size. Smaller predators could only bore through thinner shell plates or skeletons, so they stuck to those smaller prey animals. While larger predators derived more energy from going after the larger prey animals that they could handle best. This demonstrates a degree of discrimination, or the ability by the predators to sense the shape and the size of their prey and make some kind of decision or determination on whether to attack or not, and where. This adapted ability is also seen across species. For example, Claudinidae fossils are often found alongside cyanotubulites, a similarly immobile, cylindrical, filter-feeding species that lived on the seafloor, and also produced a mineralized skeleton. Although the cyanotubulites were longer, narrower, and more flexible, with a softer skeleton than the Claudinidae. These cyanotubulites are relevant because they live right next to the Claudinidae at the same time, often showing up in the same fossil beds, even right next to each other in the same pieces of extracted rock. And yet, the cyanotubulates don't show any comparable damage from predation. They don't have boreholes like we see in the Cloudinidae. This would suggest that the cyanotubulates had some quality, some trait or feature that discouraged predation. Perhaps their bodies were just too narrow, and the available soft biomass was just not nutritious enough to justify the effort of drilling through it, even if their exoskeletons were a bit softer. Or, alternatively, perhaps they were covered in soft tissue appendages that had thorns or stinging cells or some other type of defensive weapon. In any case, whatever the cyanotubulates had that protected them, this too demonstrates a degree of analysis and decision-making among the early predators in the Ediacaran. The predators had adapted the base physical equipment, the hardened mouthparts and the appendages, to scrape or drill their way through mineralized shells. But even more importantly, they had adapted a neurological system that allowed them to consider the available pool of prey animals, calculate the optimal combination of nutrient reward versus effort required to get it, and then make decisions on which individual prey animals in the area were the best targets. Now this may sound simple, like, well, obviously you would do that. That's pretty straightforward, basic stuff, right? But it represents a profound degree of intelligence and sensory sophistication in these really early animals, in these early predators. Okay, let's pause for a moment and take note of where we are. We've just explored all three of the major late Ediacaran biotas. The Avalon biota, with its preponderance of simple sessile filter feeders. The White Sea biota, with its surge in biodiversity, including the proliferation of creatures capable of free movement, as well as the emergence of predators. And lastly, the Nama biota, which struggled to maintain biodiversity in the face of increasingly adverse conditions. Now the question becomes, what were these adverse conditions? What was going on in the late Ediacaran to blunt this great pulse of Precambrian biodiversity? There are several factors that we can examine, but perhaps one of the most relevant biological factors were the microbial mats. Recall that these microbial mats had existed in pristine condition for 1.5 billion years before this, and they would have been heavily disrupted by the presence of all of these macroscopic organisms. In what was, to the microbial mat ecology, the evolutionary blink of an eye, there's suddenly dozens and dozens of creatures that are puncturing through the mat to anchor themselves to the seafloor, or who smash and roll across the mats, like the Thectardus, or who roam across the surface looking for food, like Proarticulata and countless other organisms like it, who left behind them a trail of microbial devastation, a canyon-like trackway left behind in the mud-like microbial mat. We are seeing more and more the breakup and fragmentation of these microbial mats as macroscopic life diversifies and adapts to the seafloor environment. This ongoing degradation of the microbial mats is a slow but much more fundamental, a deeper ecological shift that's going on in the background, while other faster-paced, more action-packed evolution of the multicellular eukaryotes doing all the damage is going on in the foreground. For example, in pretty much all of the White Sea and Nama fossil beds, we see a surge in ichnofossils, or ichnogenera, which are genera that we only know about indirectly. We've never found an actual specimen, an actual individual in these genera, but we've found the things that they left behind, like trackways, or burrows, or egg cases, or some other kind of evidence that reveals the presence of some otherwise unknown creature. These are ichnofossils, and the implied organisms that made them are organized as ichnogenera. There are several groups that are only known for the tracks they make through the microbial mats, which have been fossilized with deep gouges carved out of them, kind of like a tiny version of a glacier carving a trail through a rocky terrestrial landscape. In the Nama biota, there's an ichnogenus called Diplocraterion, which is known only from its burrow. These are particularly unusual because they're vertical burrows, sometimes looping and forming a U shape. There are fascinating features in their burrows which suggest that they were being constantly dug deeper into the sediment. Further analysis reveals that the surface layers were being eroded away over time, and the Diplocraterion had to regularly dig a little bit deeper to ensure that their burrows were deep enough and not too exposed to the surface. Some, albeit fewer, fossils show signs of the opposite process, where material was deposited onto the surface in which they built their burrow, requiring them to dig up a bit and fill in the deepest part of their burrow so that they don't get buried too deep and stuck in the mud and silt. It's a balancing act, and we can see the evidence of this corrective digging along the inner surface of their tunnels. We've also found actual fossilized organisms and their trackways, like the Acaria wariutia from the Ediacaran Hills in South Australia. I already mentioned these primitive but important bilaterians from the White Sea period, but it's worth saying again that as slug or worm-like creatures roaming across the seafloor, they created distinctive trackways through the mud and the microbial mats. The takeaway point here is that the increasingly busy surface of the seafloor meant that the microbial mats were no longer a viable, stable habitat. They were being too disrupted by grazers and scavengers and nearby filter-feeding organisms that punctured the mat like a hot iron in the snow. Their time had come, and by the end of the Ediacaran, they were largely destroyed. Of course, some isolated patches of microbial mat still existed in some places, but they were a fraction of what they once were. The 1.5 billion year legacy of microbial mat ecological dominance had come to an end. Now, in the late Nama and heading into the Cambrian, the microbial mats were reduced to small fragments and patches, just bits and pieces, in remote and dangerous environments, places where these bioturbating grazers and other mat-destroying organisms were less likely to reach them. The collapse and the retreat of the microbial mats had dire repercussions for the rest of the Ediacaran biota, most of which had evolved and diversified within an environment where the microbial mats were still intact and healthy and functioning. In effect, the larger, newer organisms had so thoroughly disrupted the microbial mats over the last few million years that they basically destroyed their own habitat. Like a swarm of locusts eating all of the vegetation in an area and then starving to death en masse, the mini grazers of the bacterial mats soon found themselves desperately short of food, desperately short of places where they could comfortably live and move around. The bacterial mats in which they once crawled and absorbed food had fragmented and shrunk, and they couldn't support the populations they once did. And so, the mini Ediacaran grazers began to diminish. Their populations shrunk, many of them starved to death. And in turn, so too did the animals that preyed on them, many of these new predators that had just arrived on the scene. Animals that were capable of movement fared a little better than those stuck in one spot, as they were able to move around and seek out better or more preferential environments. There was more going on here at the end of the Ediacaran than just the competition among organisms. The world itself, the entire planet, was undergoing significant geological and environmental shifts, which altered the marine ecosystem in ways that many Ediacaran creatures struggled to withstand. For example, we've found evidence of drastic fluctuations in the concentration of certain gases in the atmosphere and in the oceans, including oxygen. During these brief, oscillating periods of anoxia, animals that were capable of movement would have been able to seek out higher ground, i.e., shallower water with relatively higher traces of oxygen, or surface-dwelling microhabitats that were locally enriched in oxygen, like what you might find, almost like an oasis, among the last of the scattered shreds of the microbial mats. But it wasn't all death and destruction. As the grazers and the burrowers and various ground-based animals roamed around and made trackways and tunnels, yeah, they were ripping up and destroying the microbial mats, but they were also ripping up the seafloor itself. Their accumulated tunneling and burrowing and crawling was like a slow-motion biological tilling of the field, so to speak. The seafloor sediments, which until then had been compacted for millions or maybe even over a billion years under these thick, heavy, mud-like microbial mats, now they were being loosened up a bit. There was more water flow, so there was more available supplies of oxygen and nutrients below the surface. And on that note, the destruction of the microbial mats also led to the disappearance of sulfur-metabolizing anaerobic bacteria, which lived in communities along the anoxic interface between the bottom of the microbial mat and the surface of the mineral seafloor. This destruction of the mats and the bioturbation of the now exposed seafloor actually made the benthic habitat much more hospitable, much more oxygenated. Just not for most of the Ediacaran biota, including those sulfur-metabolizing anaerobic bacterial communities. Around the end of the Nama time period, around 542 million years ago, things had really started to change. An evolutionary event of titanic importance was happening, known as the Cambrian explosion. Now, even though we call this an explosion, it wasn't a single, sudden event, so to speak, but it was rather a period of extremely high diversification across multiple clades, and it lasted for about 15 to 25 million years. This event occurred because the Ediacaran biota had accumulated enough adaptations that now complex life was really quite sophisticated. There were all manner of basic traits, including skeletal components, shell plates, specialized mouth parts, all kinds of organs and metabolites and venoms, and a broad array of behaviors and life strategies. Think of the early Ediacaran biota, and I'm talking about like early, like Avalon and before the Avalon, where most of the animals or animal-like organisms that were alive then were just sessile filter feeders. If you don't have any organisms that can move, you're not going to have predators, or bioturbating grazers, or populations that migrate or roam around in search of food. You're not going to have anything swimming around in the water column. This is a quiet, low-complexity ecology with few specialized niches beyond static filter feeders adapted to various water depths or substrate types, and of course the tiny niches in the microbial mat. Now imagine the later Ediacaran biota, which had sessile filter feeders as well, but also had mobile animals like the grazers, the burrowers, and those that swam freely in the water column. This is a higher complexity ecology that can establish a food chain with multiple layers of predator and prey, with lots of evolutionary pressures to drive diversification in a bunch of different directions, in what is already a community that's filled with many diverse forms. The Cambrian explosion is the result of this increase in ecological complexity reaching a critical mass, or a threshold of sorts, where there was enough organic Play-Doh for evolution to really start having some fun. Imagine you're a little kid and you're playing out in your backyard, out in the woods or something. If you have a rock, you can play games with the rock. You can see how far you can throw it or something. If you have a rock and a stick, well, now you can play more games that you couldn't play before, like throwing the rock up in the air and trying to hit it with the stick. Now imagine all of the games that you could play if you have lots of rocks and lots of sticks, but also new stuff like a bucket of water and some water balloons, a bunch of rubber bands, some scotch tape, a Nerf gun, etc. There's a ton of fun stuff you can do with all of those things, playing as a kid out in the woods. And this is an analogy for the surge in biodiversity that's seen at the start of the Cambrian period. In other words, all of these features and traits, like all of these body parts and metabolites and venoms and lifestyles like behaviors and body forms, all of these different traits and features that had been evolved in the Ediacaran enabled life and evolution to play a large number of games in the Cambrian period. There were all of these different traits and features for evolution to play with, to alter and modulate in some way, to adapt to new environments or changing environments. And when all of this accumulating biodiversity throughout the Ediacaran reached a critical mass, and it led to this this massive second generation of evolutionary experimentation with the animals, that is the Cambrian explosion. As a quick side note, the reason that we haven't seen another comparable explosion of diversity in the hundreds of millions of years since the Cambrian is, for many reasons, because competition is too intense. The ecology is saturated. Instead of creating animals with new combinations of feeding strategies, movement styles, and habitats, creating new ecological niches like what we saw in the Cambrian, life after the Cambrian had explored pretty much all of these combinations and created all of the available ecological niches. Any new diversity would not necessarily be creating new niches, but would instead be competing for space within pre-existing niches. In other words, the huge increase in diversity seen at the dawn of the Cambrian was a one-time thing, contingent on the very specific ecological and evolutionary realities of the time. A similar event is unlikely to ever be seen on Earth again, barring some massive extinction event that resets life on Earth and forces multicellular animals to begin anew with whatever simple creatures happen to survive. I should clarify that while this is a scientifically plausible explanation, it's also a little vague. In reality, the reason the Cambrian explosion happened when it did, and why something similar hasn't happened since, is a topic of considerable debate. The explanation that I just gave you about competition being too intense and ecological niches all being saturated, filled up, that explanation is simply one of the more popular and straightforward ideas, but it is by no means an exclusive or exhaustive explanation. It's just one of many ideas that explain at least part of the overall phenomenon. Anyway, the Cambrian explosion produced the ancestral groups of virtually all modern phyla that hadn't already emerged in the Ediacaran, and it saw a radical transformation of the planet's biosphere. The Cambrian explosion produced the first unambiguous echinoderms, the first true arthropods, countless varieties of worms, and it saw the emergence and diversification of the chordates, including the tunicates first, and later, about 518 million years ago, the first unambiguous vertebrates in the form of jawless fish from the clade Agnatha. For the soft-bodied Ediacaran Biota, this changing environment, including the rapid influx of new kinds of animals, would have displaced them, reduced their numbers, and just in general made life more challenging because of all of the competition for resources and the subsequent changing environments brought about by the introduction of these new creatures and these new ecological dynamics. It's sort of like if you lived your whole life in the same neighborhood. Lots of people move out, but also lots of people move in. Houses and complexes are built. Some are torn down and built anew again and again over the decades, so that the neighbors, the buildings, the trees and the parks that you knew as a kid are all long gone. And the neighbors and buildings and public amenities that you see around you today are totally different. And they may be different than what existed a decade before, and that might be different than what existed a decade before that. And depending on how long you've lived in this neighborhood, you might have seen multiple iterations of things come and go. People, buildings, parks, and nature, all of it. It's an experience that can be kind of disorienting, or sad in a nostalgic kind of way. Or it may be one of those things that you make a little mental note of one moment, and the next moment, when a few brain cells have had a chance to think about it for a second, the hefty realization of your own age and mortality collapses on you, and it draws out this sense of existential dread. This was, perhaps, the collective experience of the Ediacaran Biota around 542 to 539 million years ago, which would have found its habitats enriched with new generations of animals, with new features and new body forms that had never been seen before. The ecology and the food chain had massively increased in complexity, and much of the Ediacaran Biota found itself ill-suited to compete. Although, truth be told, we don't know for sure why much of the Ediacaran Biota disappeared. For a few years now, there's been 3 competing ideas about what happened, and only in the last decade or so has evidence started to build up in support of a coherent working hypothesis. One of the 3 ideas to explain the disappearance of the Ediacaran biota is that environmental conditions slowly changed to the point that the fine detailed preservation method that recorded much of the Ediacaran biota was no longer possible. Like, maybe there was a drop in rainfall and subsequent erosion that massively reduced the frequency of fine particle mudslides into shallow water habitats. Perhaps there was a lull in volcanic activity that cut down on the levels of fine ash particulates in the air and in pyroclastic flow that would eventually build up on land and in shallow water habitats. There are several mechanisms that have been proposed, but all of them have been rejected for one reason or another. Basically, no matter how you look at it, there's lots of evidence against this idea, as all of the various geological mechanisms that could fossilize soft tissues with this fine detailed preservation, they all continued to exist. They all continued to create fossils, long after the end of the Ediacaran. It's just that these later fossils had a sudden and distinct lack of Ediacaran organisms. The second, and perhaps most intuitive idea here, is that there was a great mass extinction event, something widespread and devastating that the Ediacaran biota simply weren't able to defend against or recover from. This idea is arguably more plausible, and there's much more evidence in support of it than there is for the taphonomic idea I described a moment ago. For example, a 2008 study postulated that a major change in ocean currents brought hydrogen sulfide-rich water from the bottom of the water column near the deep seafloor up to the top of the water column, near the surface and these shallow water habitats. The sudden presence of hydrogen sulfide in these habitats would have poisoned and wiped out many of the organisms that lived here. But there's a stronger case to be made based on a carbon-13 anomaly found in shallow water carbonate-rich sites around the world, all dating to 542 million years ago, that suggest that the end of the Ediacaran saw a brief period of anoxia, one that could have been caused by a widespread influx of hydrogen sulfide. It's possible that hydrogen sulfide and other sulfur compounds were loosened up and they leaked into the water column because of the destruction of the microbial mats. These microbial mats would have previously covered up these hydrogen sulfide producing microbial subcommunities that existed at that interface between the mat and the mineral surface, and they would have bottled up all of that hydrogen sulfide. But as the mats collapsed, and they represented not just a loss of habitat for the grazers and the burrowers and those other moving creatures, but they also released a lot of this pent-up hydrogen sulfide. All it took was a change in water circulation, a change in the currents, to sweep up massive amounts of this freed hydrogen sulfide into shallow water habitats, and it choked out a lot of the things living up there. So not only would the hydrogen sulfide have been poisonous, but these compounds would have reacted with the oxygen. They would have absorbed the oxygen, pulled it out of the water, and made the water anoxic. And it's perhaps more likely that it's this anoxia, and not so much the poisoning from the sulfur compounds themselves, that wiped out a lot of these life forms. You'll recall that I mentioned a few minutes ago the benefit of movement, which allowed animals to seek out more comfortable habitats when things got tough, like during this apparent anoxic event. Sessile animals, like the old rangiomorphs and the proto-anemones and whatnot, were doomed. They were stuck in one spot, and they couldn't escape to safer areas if their current habitat became too anoxic, or too polluted with hydrogen sulfide, or too warm, or whatever else it could be. This idea of a brief anoxic period at the Ediacaran Cambrian boundary is further supported by changes in molybdenum isotopes, in thorium and uranium isotopes, and in a temporally localized surge in organisms that thrive in low oxygen conditions, like cyanobacteria, and a sudden drop-off in the frequency of trace fossils from animals and proto-animal Ediacaran relatives. The problem is that a lot of this evidence isn't strong enough to conclusively say that this anoxic event definitely happened, that it definitely affected this and this and this geographic regions, and that it definitely killed off X% of Ediacaran life. Rather, the evidence suggests a few potentialities, all of which have been debated, all of which may have had some influence or some limited role in disappearing the Ediacaran biota, but nothing has been nailed down as a definite cause. In fact, the rapid disappearance of the Ediacaran biota that we would expect to see in a mass extinction doesn't exist. So the reason this evidence, it's suggestive, but it's not overwhelmingly strong and conclusive, is because a lot of the data that we would look for as a smoking gun just doesn't exist. There is no sudden disappearance of the Ediacaran biota, akin to the asteroid that wiped out the dinosaurs. Rather, it seems that the Ediacaran biota gradually disappeared, and they were gradually replaced by the new Cambrian biota over a period of 9 to 10 million years or so. This gradual replacement idea is the third idea behind the disappearance of the Ediacaran biota, and among all three of these competing hypotheses, this gradual replacement idea is perhaps the most validated and the most accurate. In essence, if we look at the fossil record closely and we trace when these groups disappeared, we see that there was a slow-motion biological turnover event, wherein the old Ediacaran neighbors moved out of the neighborhood, gradually, one at a time, and new Cambrian neighbors moved in. These newly emerged Cambrian organisms would engineer the ecosystem in ways favorable to their lifestyles, which often led to disruption and displacement for the Ediacaran organisms. So as this period of turnover dragged on, the rate of change accelerated. The Cambrian animals were increasingly well suited to the world they were creating for themselves, while the Ediacaran organisms were increasingly incompatible with it. What do I mean when I say that these new Cambrian organisms were ecosystem engineers? What is that? I mean, by nature of going around and doing their thing, living their life, they altered the physical or the chemical environment in a way that benefited them, but it pushed out other organisms. For example, we saw bioturbating organisms appear and increase in complexity throughout the late Ediacaran, but going into the Cambrian period, the number and diversity of these grazing, trackway-carving, crawling bilaterians increases dramatically. It would appear that many of these creatures emerged in the late Ediacaran. They were partly responsible for destroying the microbial mats, and then they adapted to crawl and burrow through a wider diversity of inorganic substrates, as one would need to do if they were tunneling through the raw sediment on the seafloor without the cushy layer of a mud-like microbial mat. By adapting to the seafloor habitat after the bacterial mats were destroyed, they were ready to compete and rapidly diversify in this new Cambrian world. We also see the emergence of true arthropods. These are thought to be a Cambrian group descended from the Proarticulata that emerged in the late Ediacaran. With their segmented bodies armored with chitinous exoskeletons, they represented a distinct and unique permutation on the tree of life relative to the rest of the soft-bodied, unsegmented Ediacaran life forms. Instead of burrowing through the microbial mat and absorbing nutrients directly, they evolved various hard parts that scratched the microbial mats and at algae, kind of like the purported proto-mollusk Cambriella, and the idea that it had evolved a pseudo-radula to scrape at algae and other yummy microbes coating rocks in its habitat. This gradual turnover idea ties in nicely to the changes in the Ediacaran biota that we observed from the White Sea to the Nama time period. In the Nama, the Ediacaran biota struggled to maintain the biodiversity that it had achieved in the White Sea, and we now know that this is because the turnover event was already happening. To put it another way, the Nama time period itself seems to be the critical juncture where the Ediacaran biota gave way to the new forms that would dominate the Cambrian. There's a few Ediacaran creatures that were able to survive the stresses and pressures of the Cambrian, including competition with the newly emerged forms generated in the Cambrian explosion. Some of these Ediacaran lineages persisted for just a little while into the Cambrian, like the spiral-shaped Cochleatina, the extremely basal annelid worm Cybelidates, the amoeboid protists of the Platysolenites, and most of the Cloudinidae that I talked about earlier. Even the microbial mats were able to persist a little bit into the Cambrian, but by 530 to 529 million years ago, these had, for the most part, all disappeared. One group of Cloudinidae, known as the Cloudina, disappeared almost right away at the 542 million years ago boundary. The Saurina was a similar type of organism, its body a 7 mm thick cylinder of stacked funnel or cone-shaped ring segments. During the Saurina's growing phase, their body was soft, but in maturity, they'd exude a mineralized exoskeleton that formed a thin protective crust around their tissues. These emerged in the early Nama time period, about 556 million years ago. They survived a little bit into the Cambrian, but by 535 million years ago, they were gone. Another creature, known as Corymbella, represents the very tail end of the Ediacaran Biota. It too has a body shaped like a long column or filament extending upwards from the seafloor. Except instead of possessing a frond like a filter-feeding rangiomorph, or funnel-shaped cups like Cloudinidae and Saurina, these Corymbella organisms possessed a long, unbranching stalk composed of interlocking angular rings. The rings are believed to have been composed of, or coated by, a layer of calcium carbonate in the form of the mineral aragonite. And at the end of the stalk, they're thought to have possessed some sort of soft tissue structure to filter out food particles from the water, like a nest of ciliated tentacles or something like that. These Corymbella creatures were even more short-lived than the Cloudina. Corymbella is known only from a slice of the rock column dated back to 543 million years ago, so in a way, you could say that they were among the youngest casualties of this great transitional ecology and the biological turnover leading into the Cambrian. Other lineages were much more successful, and they survived for hundreds of thousands, if not millions of years after the Ediacaran. One such group of hardy survivors are the Conulariida, which are incredibly ancient relatives of the Cnidarians. These were anemone-like organisms, with a mess of stinging tentacles protruding from the broad end of an inverted cone. These Conulariida emerged in the fossil record in the Upper Ediacaran, sometime around the late Avalon and the White Sea transition period, 570 to 550 million years ago. But unlike most of the Ediacaran biota, they persisted through the Cambrian explosion. More than this, they persisted throughout the Cambrian period itself. They persisted throughout the Ordovician. They survived through the Devonian, the Age of Fish. They survived the end Permian mass extinctions. And they finally tuckered out and fell into extinction in the early Mesozoic, at some point in the Triassic period, around 245 million years ago. This was about 300 million years after the rest of the Ediacaran biota had largely gone extinct. That's an incredible track record, especially compared to almost everything else that emerged way back when they did. I mean, how many Ediacaran lineages can boast that they overlapped in time with the very earliest dinosaurs? Another cool detail has to do with the conical body of the Conulariid. The conical body has a hard part on the outside that contains and protects the soft tissue. This hard conical outer surface has a four-fold symmetry. The cone isn't perfectly circular. It has four bulbous fans running down its length, so a cross-section might look sort of like a swollen plus sign, or a horizontal cross-section of a bell pepper, or something like that. A particularly old fossil suspected of being an ancestral Conulariid has a six-fold symmetry. Over time, this was reduced to a four-fold symmetry that they maintained for hundreds of millions of years. But that original six-fold symmetry is thought to be a derived trait from a three-fold symmetry like that seen in the Trilobizoans. This hypothesis suggests that the Trilobizoans may be deeply ancestral to the Conulariids and other Cnidarians, or perhaps they could be a stem group offshoot, which would be a super interesting twist. This possibility is the only way that we could say that the Trilobizoan legacy outlived the Ediacaran. Much like how the birds are legacy of the dinosaurs, the only Ediacaran lineages still alive today are those that diversified into forms that could withstand the calamities and evolutionary pressures of natural history. I'm thinking, of course, of the sponges, the ctenophores, the cnidarians, and all the rest of the basal eumetazoans that still have descendants swimming or filter feeding in the ocean today. Everything else, all of the fantastic and unique forms of the Ediacaran biota, disappeared with the advent of the Cambrian. The rapid diversification of life produced increasingly sophisticated forms with greater capabilities and ecological impact. For example, consider the sophistication of muscle tissue, which we saw initially as simple sheets of contractile cells that wrapped around the body of cup-shaped anemones, and compare this to the much more organized systems of skeletal muscle that we see in the earliest vertebrates. This represents a massive jump in tissue complexity and capability. It's easy to see how the Mark I Ediacaran biota were no match for the Mark IV, Mark V, Mark VI animal body types emerging in the Cambrian. The fossil record backs this up, showing the Ediacaran biota slowly being pushed out and replaced over many millions of years by these gradually better adapted and more capable Cambrian-era creatures. This is a darkly ironic fate for the Ediacaran biota, as they set the stage and built the evolutionary groundwork upon which the Cambrian explosion could take place. Just as the Ediacaran grazers destroyed the microbial mats and thus their own food source and habitat, their Cambrian descendants, in turn, destroyed them by outcompeting them, changing their habitats, and gradually replacing them in ecosystems around the world. The Cambrian biota, of course, would go on to dominate the planet, filling the oceans and covering dry land with an incredible menagerie of life forms. And all of them contain a bewildering complexity of biochemistries and deeply derived body forms, shaped by 500 million more years of evolution than the Ediacaran biota ever had. The Cambrian biota obviously overshadowed their Ediacaran forebears, and they take up virtually all of our attention today when we talk about the ancient past and deep evolutionary history. And yet, the Ediacaran biota retain a kind of humble elegance. These were the creatures that lived in a quieter world, a simpler world, with more rudimentary communities and basic food chains. They enjoyed millions of years to themselves, quietly filter feeding and exploring this biologically undeveloped world. They were not large, nor were they very complex, but they did serve a crucial purpose. They were the first generation of evolutionary experiments towards an animal-like entity, a multicellular heterotrophic organism of tremendous size relative to all of the microbes and the fungi and the plants that came before. This experimentation naturally produced countless bizarre and inexplicable forms that disappeared from the fossil record just as quickly as they arrived. Some forms lasted longer, and a particularly successful number of lineages were able to spawn superior forms that gradually replaced their predecessors, leading to the Cambrian explosion and the diversity of life that we know today. None of this, none of us, would be possible without the Ediacaran biota. These were the organisms that came before us, settling the evolutionary landscape by finding optimal forms and developing key traits, like mineralized skeletons and mouth parts and bilateral body symmetry. And these fundamental traits that survived the rigors of evolutionary pressure were passed on to be further modified by evolutionary tinkering in perpetuity. And here we are, some 540 some odd million years later, having achieved a level of complexity and diversity that the Ediacaran biota would never know and could never even dream of. And yet, it's a level of complexity and diversity that they were, in a way, ultimately responsible for. We would not be here in the way that we are without them, and our entire biosphere would not be what it is today were it not for those first forays into animal multicellularity, those first biochemical superstructures that freely explored what it was like to exist in the world at the mesoscale. Those evolutionary lessons have shaped all animal life on Earth ever since. That is ultimately the legacy of these mysterious, cryptic, almost symmetrical, soft-bodied creatures of the Ediacaran Biota. This has been episode 100 of the Biologic Podcast. If you like this, then subscribe to the channel on YouTube or through whatever podcast hosting service you use, so you can get all of my biology content as soon as it's released. If you want to donate to help support the show, then check out the Patreon page, where you can pay as little as $1 or $2 a month. I release about one show a month, so that's $1 a show. Now, most shows aren't super long, special episodes like this. This was a special occasion. It was episode 100. I was going to go all out. But this is also not going to be the last special episode. There's going to be more special episodes like this in the future, in the near future. So it's definitely worth it for you to subscribe and support the show. In the next episode, we'll return to the early metazoans and explore the lifestyles and anatomy and ecology of the mini cnidarians, like jellyfish and sea anemones, and the hydra and siphonophores. So definitely subscribe for that. It's going to be a super cool episode. However you support the show, whatever you do, you guys make this all possible, including special episodes like this. And as always, thanks for listening.